From Microbe TV, this is Q&A with A&V. I'm Vincent Racaniello, and joining me tonight from Chevy Chase, Maryland, Amy Rosenfeld. Hello, Amy.
Hello, Vincent. How are you today?
Not bad. And how are things in Chevy Chase?
Disgustingly humid.
Is it raining down there?
No, it just is disgustingly humid.
It's been raining here for a few days. Happy New Year, everybody. Happy New Year, Amy.
New Year. Happy New Year. Did you have a good holiday?
It was okay. I'm not a fan of holidays.
I know, but then you had a birthday, so happy birthday.
Thank you. Yes, January 2nd. I left the 6th decade of life behind. Should I show that picture that I made, do you think? Would that be cool?
Yeah, for sure.
It's a really nice picture. I spent a long time doing it because I had to get the lighting right and the angle and this. It took about an hour to do a passable picture for Instagram. Isn't that something? So anyway, let me try and get it up here. Library. Let's see, where would that be roughly? There's so many versions. It's the one with the blue glasses. There we go. Sorry, folks, for the delay here, but you'll like it, I believe. Okay, here we go. Where's Vinny? There he is. How's that?
Looks great.
Boom,
You
boom,
and the
boom.
virus.
Me
You and the
my
virus.
It's just perched on my hands. And it was an Instagram post that many of you probably saw, leaving my 6th decade of life. And who knows what the future will bring, but it will have my viruses and, of course, all my podcasts. So there you go.
Super exciting.
And thanks, everybody, for your birthday wishes. And that, I have this confluence of Christmas, New Year's, and birthday. It's overwhelming, Dr. Rosenfeld. It's overwhelming.
I can understand. So where are you going to go on vacation to recuperate?
No, I'm not going on any vacation. But what I'm doing, Amy, is back to the incubator and doing the things that I like to do. Okay.
That's great. And where's my ad for my job?
You have to send it to me. And we'll read it this Friday, okay? But email it to me so I remember. Here,
again.
Amy's looking for a technician. Here you go. You could email her there, amy.rosenfeld@fda.hhs.gov. But we will do an ad on Twiv again. I wanted to thank the moderators tonight. We have Les, we have Steph, we have Vanity, we have Tom, and we have Frank all here tonight. Thanks all of you for coming by. We have 230 people already here, and we're going to answer your virus questions. But I wanted to show you first. Remember the Amy papers, right? Where are the Amy papers? There they are.
Do you even update them?
I did today, yes, and I picked a bunch that we're going to quickly zip through right now. Okay.
Do you got a zip car?
Oh, Vanity's birthday was on the 30th. Happy birthday, Vanity.
Happy birthday.
And wasn't Fauci's birthday on the 24th?
Yep.
All right, so
82.
82.
We need a zip car. We need a little zip car icon running around when you say we're going to zip through things. We need a little zip car emoji.
And folks, by the way, everyone's wishing me happy birthday. Thanks so much for your wishes. I get a lot of... when I heard that Tony was 82, and I think you told me.
Yes, I did.
I immediately felt better because I figured my next 20 years, if I live that long, are going to be garbage. But not, apparently, because Tony's been running around, so...
Tony is a power walker.
I am, too. I treadmill 3 times a week. And
That's
I walk
good.
outside once a week, too. And my son, for my birthday, gave me a fitness program.
That's great.
He's going to evaluate me, and he's going to tell me what to do in addition to the
treadmill.
And I spoke with Steph this week, and she recommended a few things for me, one of which is a few times a day just to close my eyes and meditate. Just listen to your breathing, focus on some sounds, just relax
Okay.
for a couple of minutes. So I did it on the train this morning, standing up. I had the sun over the meadowlands shining in my face. I closed my eyes and I just listened to the sounds, and it was very relaxing. Right, so I thank the moderators. Let me show you a couple of papers, and Amy, you and I can
I'm
bring
still
in on
waiting for
this.
the zip car.
Wait, hang on. There's no zip car, dude.
Well, you need
There's
a
no
zip
zip
car.
Where is this thing? Okay, here we go. Are you ready for our little thingamabobber? Screen share is on. I have to make sure this is working. Can you see it? Okay,
Yeah, I see it.
now this
The
paper...
alarming antibody evasion properties, yes.
Yes. So this is an unfortunate title, Alarming Evasion Properties. I don't think there's any room for that kind of a title in a scientific journal. All right, but this is out of David Ho's lab. And this is about the new subvariants of Omicron, BQ and XBB. They have increased ability to evade neutralizing antibodies. And they also are resistant to bebtelovimab. So now we have no monoclonals for therapy. And they say this is horrible. But the real question is, and I'm going to turn this on so I could look you in the face, does it matter? Are the vaccine-induced antibodies, T cells, etc., still protective? And there's no information on that. I think they probably are, right, Amy?
Well, are there more and more people dying and being in the hospital for severe disease?
Not as far as I know. So that's the kind of study you need to say that these variants are concerning. Not this one, an in vitro neutralizing antibody test. So, and this has been going on throughout the pandemic, as we know, Amy, right?
Yep.
Okay, so that is screen share back on. We're going to go through these zippy. Here's an article from the Wall Street Journal that Amy sent because I don't subscribe. Are vaccines fueling new COVID variants? I think this is absurd.
It's insulting.
Immunity fuels variants. It selects for variants, any kind of immunity. And maybe my understanding is so few bivalent vaccines have been given out that probably doesn't make a difference, right? Although the title is just vaccines in general. I don't know what the point of it is. We need vaccines, otherwise people will die. So why don't you just say, is immunity selecting variants? And that would be a stupid headline, right? So they have to make something kind of controversial. You think?
Well, if you made a headline of immunity selects variants, what is the general public going to do with that headline? You can't get rid of your immune system.
No. And
right.
You can't get rid of vaccines, right? Because if you don't have vaccines, people will die by the hundreds of thousands.
Well, so right. So I don't really quite understand. I mean, I don't really understand her point. So
no, I don't either.
And it's not even immunity that selects for variants, right? Viruses make mutations every replication cycle.
Of course, but then the ones that evade
there's
some that
are selected for, but they don't all evade immunity.
No,
they have other properties.
Of course not.
So the other thing is I find it offensive that this idea of neutralizing antibodies, whether or not they're protective or not, is only about whether or not it basically binds the virus in vitro. And that suggests that they are uncreative to think that the FC portion or the other portions of the full antibody have any role, and that they're mutually exclusive. And they're not, just like I said last week. They're not. I'm sure that there's an FC component to the antibodies that we think bind and neutralize.
All right, the next one, SARS-CoV-2 infection and persistence in the human body and brain at autopsy. So in this paper they looked at autopsies on 44 patients who died, and they did PCR to look for viral genomes. And this paper is just, this infection and persistence is wrong because they cannot say that by looking by PCR. And look what they say here. We show that SARS-CoV-2 is widely distributed predominantly among patients who died with severe COVID. Okay, SARS-CoV-2 RNA, and that virus replication is present in multiple respiratory and non-respiratory tissues, including the brain, early in infection. No, you cannot say that virus replication is present in multiple tissues, right, Amy?
Right.
By a PCR assay.
Right.
Come on, neurologists, or whatever you are.
They're not virus people.
They're not virus people. You and I are virus people.
Yes, I'm virus people.
Okay, the next one. COVID, this is from, where is this?
Nature.
Article? Nature. I think it's open access, so you can check it out. COVID drug Paxlovid was hailed as a game changer. What happened? What they say basically here is that, we still have a lot of deaths because we're not using COVID. A lot of countries don't get it. And in the United States, not everyone gets it. Look, physicians have prescribed the drug in only about 0.5% of new cases in the UK and 13% in the US. Even doctors have reported serious difficulties in helping their family members to obtain COVID. And part of it, they say here, is this stupid issue with COVID rebound, right? It's absurd. Anyway, it's an interesting article because this is a good drug. It does very well. Okay. All right, 2 more. It's actually quite good. WHO estimate of excess mortality associated with the pandemic. So excess is in addition to people dying of COVID. What else is going on? And this is hard to estimate because, as you see, we have variations in testing access, differential diagnostic capacity, inconsistent certification of COVID as the cause of death, which, Amy, you were talking about last week, right? Often hard to determine. Anyway, they used some statistical methods. They estimate 14.83 million excess deaths globally, 2.7 times more deaths than the 5.42 million reported as due to COVID for the period. So probably some of these are COVID, right, Amy? They're just not properly reported.
I would assume so.
But others could be, like you said last week, like a heart attack caused by COVID, but not reported as due to COVID, right?
Yeah, for sure.
And then one more. This is quite interesting. This is a bioRxiv preprint. This one is telling us that the XBB variant is a recombinant of 2 other Omicron subvariants.
Shocking. Oh my God! An RNA virus went through a recombination event. I might need to be
co-circulating lineages during the summer of 22 around India. They recombine, and the recombination breakpoint where the 2 RNAs joined is in the spike, in the receptor binding domain of spike. And each region of the recombined spike from either virus confers immune evasion and fusogenicity. So this is, they say, the first documented variant increasing its fitness through recombination rather than single mutations. I would say that we're aware of, right? Because maybe recombination might have been involved in some of the others.
Well, I'm sure.
Anyway, so those
oh my God, can you believe that an RNA virus went through an RNA virus recombination event?
Yeah, they
have you ever heard of such a thing?
Does poliovirus recombine, Amy?
Of course.
I bet it does. All of your beloved picornaviruses, or I should say enteros. You're not a picorna fan, right?
What do you mean I'm not a picorna fan?
Do you like picornas, like cardioviruses?
I use EMCV.
Yes, they recombine. In fact, when you get OPV, within a few weeks you shed recombinants, right, Amy?
Yes.
Very cool. Anyway, this is the Amy Papers. That's this week's digest from the Amy Papers. There are many more. They're microbe.tv/the Amy Papers. And these are papers that Amy sends me and Daniel, and sometimes just me. And I put them up there. And you guys can see what she's thinking about. You can get into Amy's head.
Don't do that. Not. It's a mess up there.
Don't do that. Okie dokie. Let's get to the questions from our listeners or participants here. Polio Pete is here. Hello, polio.
Where's
Pete.
my cookies?
In the New York Times 7-day average last year at this time was 50,000 new cases. This year, 7,500. Yeah, it's very good.
So it's 8 times less.
So what's responsible for this?
Vaccines and immunity.
Vaccines and immunity, yeah. Yep. Would you say we are endemic now, Amy? Would that be fair to say?
Not quite yet.
What do we need to do to get endemic?
We need to have more immunity worldwide, globally.
Yeah, because China's having a huge epidemic, right?
Yeah.
Amy, here's one for you. Is the year-to-date new case count skewed because of less testing?
I'm sure it's no more skewed than it was previously, so that probably you can subtract the idea that it's skewed, and it's probably close to what it should be.
Okay.
Because we didn't test at the beginning. We didn't really test so well in the middle. We don't test so well now. I mean, come on.
No, we don't.
So if we missed 20% in the beginning, if we missed 50% in the beginning, then we missed 45% in the middle, and now we're missing 43%. What's the difference?
All right, Patricia wants to know about Lyme vaccines. There isn't one at the moment, but there are clinical trials underway. It's in phase 3. One of them there's 2 companies, Valneva and Pfizer. I guess they've done it together. A multivalent protein subunit vaccine that is targeting the North American and European strains of Lyme disease bacterium. So it's in phase 3. The other one didn't work that well, so it was taken off the market. Yeah, and a lot of people are getting their stickers. That's great. I'm glad to hear it. And some of them like my signature. I signed all the backs of the envelopes, Amy. Do you know that?
I've heard the rumor.
You've heard the rumor?
I've heard the rumor that you're freely giving out autographs. I've heard the rumor.
Yeah, well, I'm not so famous, so it doesn't matter. Here,
one day you will be.
This is what the letter looks like on the back. I do a nice signature, and
it's great.
Glenn Ross says, "Why don't you make an O?" Well, that's part of the style. I don't make the O. So if you got your sticker in this envelope,
doesn't he understand? Everybody needs to be different.
Oh, by the way, I'm cleaning out my office here, and I threw them out. Never mind. And by the way,
wait. Did I hear the right words? I threw it out.
I threw it out.
I threw it out.
Here's a garbage can full of stuff.
You use those words. I threw it out.
Look, throwing out stuff. Throwing out a lot of stuff.
Oh my God.
Yeah.
Luckily, I'm sitting down because a girl could hurt herself.
What was I talking about? All right, it's gone.
You were looking for something that you threw out.
I had an old pair of glasses. I was going to show everyone what I used to look like, but I threw it out, so you're not going to see it. Not going to see it.
Oh my God.
So there was a question here. No question. Okay. Is anyone having trouble with the super chat? I haven't seen one yet, and our friend Kip and Laura from San Francisco said he's not able to donate. Yeah, maybe it's YouTube. Yeah, because Patricia said it's not possible. Okay, we'll come back next week and try again, I hope. Or you could go to Venmo, right? I don't have my phone, unfortunately. I could give you the QR code.
You have a QR code?
Yeah, Venmo gives you a QR, so you can just hold it up, and someone who wants to give you money can do it, scan it, and then you got their thing. It's really cool. It's very cool. Okay, here we go. In long COVID, where could infected cells be located? Could the virus proteins get in the blood or brain tissue? And if so, is there any particular danger if this happens?
Well, it could be in the blood, but it's not in the brain.
I think that long COVID is an inflammatory disease. I don't think it has anything to do with virus replication. Would you agree with that, Amy?
I would agree with that.
You know,
But virus does persist. RNA fragments and proteins do persist, according to Diane Griffin, for RNA viruses.
yes. So they could be
So
triggering
it could
inflammation,
be
right?
that, yeah.
Okay.
But you could find them.
For sure. So the cells could be circulating, as Amy said. The proteins, RNA, could be circulating. But, it doesn't happen in everyone. So certain people are prone, and we don't know why, particularly. Claire, for a 40-year-old healthy person with 2 shots of an activated vaccine close to 2 years ago before first infection, how many shots after infection will be solid for the breadth of antibodies, intervals? 2 people just donated money, so it's working. 2 shots and then infection. I think that's good to go, right, Amy? For a 40-year-old. You have 3 doses, basically, and you're good. Amy and I both have 3 doses of vaccine, no more, no less. So that's all you need. Has anyone identified organisms that are capable of eating and metabolizing viruses? Well, yes, you're referring to a paper that came out, and let me show you the paper. I have it actually queued up here. What do you think about that, Amy? Look. Where is this?
I don't know, but I see the microbe TV.
Consumption of viruses returns energy to food chains. It's a PNAS paper. And David Dunnigan is from the University of Nebraska. He emailed me a few weeks ago, and he said this was coming out. They basically show that plankton can subsist. Small protists can consume viruses but can grow and divide, given only viruses to eat. Now, I'm going to ask you a question, Amy. If you could only eat viruses for your meals, which viruses would you eat? Enteroviruses? And they would go right to your gut where they belong, right? What virus would I eat? I guess I would have to eat poliovirus also, since I worked on it for so many years, right?
But Mentos don't always go to your gut. 68 and rhinos go to your respiratory tract. Not always going to the gut.
I think I'm going to have one of these people on to talk about it, right? Since Dave did email me.
For sure.
Okie doke. Oops, that shouldn't be there. So, and DGH, that was a nice cue to talk about that a little bit. So there's a Bay Area storm, huh? Did you hear about this?
Yeah, I talked to Katub yesterday, who lives in San Francisco. He was telling me.
All right. Do you still develop natural immunity after taking Paxlovid?
Yes.
You do?
Yes.
I thought it was blunted.
She didn't ask if it was blunted. She just asked if he developed immunity.
Okay, you're right. But you develop some kind of immunity, but it's most likely blunted, right?
Could be.
I think there was some papers that we talked about. So, yeah, if you've never been vaccinated and you got COVID and then you took Paxlovid, your immunity is not going to be as fully developed as if you had gone through the whole infection. Yeah. Thanks, everybody. How does the coronavirus envelope proteins interact with human PALS1 determine pathogenesis, and why is it hard to study the EP in a lab? The envelope protein. All right, first of all, Amy, what is this PALS1?
I don't know.
Well, let's look it up. E protein interactions with PALS1. Yeah, there it is. The SARS coronavirus E protein interacts with PALS1. The C-terminal PDZ binding motifs, blah, blah, blah. They determine the crystal structures of this thing bound to E protein. But what does it mean, right? What does it mean? I don't know if it means anything. What do you think?
I don't know.
Yeah, I don't see any. Oh, everybody's donating now. Thank you very much, folks. DGH, I don't know that we know that it means anything. Just because proteins interact doesn't mean that has some role, right?
Right. It's hard to study because I believe it's a dimer in a membrane. It's hard to purify and stuff. I know because somebody I know is trying to do it.
And Amy will tell you that many people have made a living during this pandemic looking at protein-protein interactions that are of no consequence. I think that's a fair statement, don't you think, Amy?
Yes. It's beyond a fair statement considering the fact some of them have gotten very large amounts of money to do it, both privately and from the government, and it pans out nothing. Because at the end of the day, you have a ton of data and you guessed wrong about which one to follow up.
Amy, do you have any opinion on the paper showing declining efficacy of mRNA with successive boosters?
I didn't see the paper.
So I would guess that the efficacy they're talking about is induction of neutralizing antibodies. And I recall this paper saying the more you boost, the less increase in breadth that you get in the antibodies. So
Wait a minute.
It's not surprising.
Wait a minute. It's two different. You just said two different things.
Mm-hmm.
Okay. So decline of efficacy of the mRNA. So is efficacy defined as the diversity of the antibody repertoire, or is efficacy defined as an increase in the actual number of antibody producing cells?
I'll bet that they look at neutralizing antibody titers.
Well, then if they did that, then it would go up. It always goes up.
But the breath
they always go up,
right?
but well, you can't measure breath by that assay. The thing is that people don't understand that the start point of the line is different. So no matter how many boosts you got, it always goes up the same amount, basically. The rise of the line, the slope of the line, which is rise over run, is basically always the same.
I'm trying to find the paper.
People just get confused at the starting point.
I'm not finding it, so that's what I think is going on anyway. When are we getting the oral remdesivir mentioned in this article? Well, it's just in phase 3, so it has to be submitted for EUA, I presume, right, Amy? Is that how it works?
I don't know.
And that could take months.
I don't do those kind of things.
That could take months. But you do work at the FDA, right?
I do, but I don't do those kind of things. It depends on how fast the company moves. And I don't know. Maybe the company is not particularly speedy.
Well, they should move quickly if they would like to make more money, right?
Well, this is a separate question because this boggles my mind that they can't figure out how to make monoclonals then against the variants.
Yeah. Well, I think to answer Ian's question, it's going to be some months at least. A lot of people would like to have oral remdesivir because they can't take Paxlovid for various reasons. So hopefully we get it soon. Raz wants to know, Amy, how can SARS-CoV-2 not have a viremic phase and yet RNA is found in neurons, brains, testes, etc.?
So the virus gets ingested by phagocytosis cells, including macrophages and monocytes. It gets brought to the lymph nodes, and then those cells move around the body.
So this phagocytosis occurs in the respiratory tract where the bulk of RNA virus replication is happening, right?
Mm-hmm. It's not specific. It's non-specific. Viremic means you can find infectious virus.
I think the participants should see this, right? This lovely thingy. What do you think?
What do you mean lovely thingy? It's a sculpture, right?
I'm sorry, I was at a loss for words. Here.
Yeah, it's very nice.
This will blink. I don't think you like the blinking, do you?
No, the blinking is not good.
Timer fading, and then we have off. But I like this one. I made this years ago. I got these little LED
I know.
thingies. It's cool, isn't it?
Yeah, but stop blinking. It makes me dizzy, please.
Okay, I will. Thank you, Les, for reminding me to show that. Okie dokie. What do we have here? Yes, Amy had offered the position, but then they decided not to come, so she needs to look again, right, Amy?
Yes.
I left my 7th decade and started my 8th. Wait a minute. 10, 20, 30, 40, 50, 60. 6 decades, no?
Maybe 0 through 1, 10 through 20, 20 through 30, 30 through 40, 40 through 50, 50 through 60,
That's 6
7.
decades. I left my 6th. I'm not that bad at math.
Okay.
But everyone's saying correct. How can this be? All right, never mind.
Technically, it is correct, but moving on doesn't make a difference.
Okay, Rach from the Isle of Man. Her favorite episodes were Breathless with David Quantiman and Eddie Holmes. Yeah, those are 2 good ones. Very good. Thank you very much. Anything progressing on correlates of immunity, not only for COVID, but other viruses. Amy, what are the correlates of immunity for enteroviruses?
What?
What are the correlates of immunity for enteroviruses? What is important to protect you from disease?
Well, when we figure it out, I'll let you know.
I thought you said it was antibodies.
It's the best biomarker that we know of so far, but it's not necessarily a correlative of protection.
Okay, got it. Got it very much. Okay. Amy, what about COVID? We don't know what the correlates is. People think it's neutralizing antibodies, but I don't think that's right. What do you think?
Of course it's not right.
It's other things.
Of course. Nobody wants to do any thinking.
Here's one for you, Amy.
Yeah, that's the car.
The zip car.
That's
You asked, you got.
Car. I know. That's the zip car. Now it just needs a little. It needs a little music and go zipping around.
Hello, Rafael from Brazil. I'm in bit in doubt with the news. SARS-CoV-2 changes faster or slower than influenza? Every day,
slower.
A doomsday
much
variant.
Slower.
We just have more infections now of SARS-CoV-2 compared with influenza. It's about 10
we
times
have way
lower,
more
right?
influenza. We have way more influenza than SARS-CoV-2 infections. The mutation rate and the variant rate, the evolution rate of SARS-CoV-2 is not on par with that of influenza. It's way lower. Okay, I talk to a flu guy every day. I know this.
Okay, so the mutation rate of SARS-CoV-2 is about 10 times less than of influenza in terms of
maybe even greater
changes. Okay.
Maybe even greater than 10 times.
The variant emergence is a different story because it's based on the plasticity of the spike and so forth, right?
No.
It's not based on the plasticity of the spike? Why not?
The variants are not a different story because we don't actually... Who's genotyping any influenza? You know anybody who's genotyping any influenza right now?
Genome sequences all the time. It's huge.
Well, I'm sure that you would find the same. You would find that HA and NA and various other places have alterations.
Oh, they do, but in terms of evading neutralization...
Well, I'm sure that they evade neutralization. I don't see any papers that look at it.
No, Amy, it's much, much slower for flu in terms of evasion of neutralization. Because you change the vaccine and it works for a year if you hit the right vaccine.
You change this vaccine and it's still working 3 years later.
Yes.
So what's your argument? So therefore your argument is flawed. Your argument goes in the direction that I'm saying it changes far less quickly than flu does.
So let me just try and rescue my flawness.
No. We don't need to rescue the flaws. So for flu, we should wallow in wrongness like we did this summer. We should wallow in wrongness.
We can't use the same words. So for flu, you get an antigenic change which causes more severe disease, and the correlate of that is reduced neutralization. Okay.
In theory. But we don't do T-cell assays or anything.
So that vaccine doesn't work. But for SARS-CoV-2, you argue that it doesn't matter. All of this antigenic change doesn't matter because you're still protected against severe disease and death by the old vaccine, right? Okay. But
Right.
and you're also saying if we did more genome sequencing of flu, you'd see as many antigenic variants as SARS-CoV-2,
Oh, for sure.
right? Okay,
For sure.
I got it.
Even more.
I got it.
Even more.
And more transmissible? Not really, because what it is they're
Which
immune
is more
evasive.
transmissible? Which is more transmissible?
XBB.
No, is he saying that the SARS is more transmissible than flu?
No. Each variant is called by the press more transmissible because scientists are
Really?
quoted as saying that. But what it is more immune evasive, so they can infect an immune population and it spreads. You have enhanced spread. Now, actually,
By this time, I don't understand how we could have more transmissive. I mean, it would
well, of
seem
course,
like
it's not. It's not. It can't
yeah,
just
I was
keep
going to say
increasing from one variant to another. It doesn't make
exactly.
any
It
sense.
seems to me like it would be like the zip car.
Yeah.
See, this is why we need the zip car emoji and the racetrack with the music. You
That's
see,
right.
this is why we need the zip car.
All right, Amy, your January 13th blog. Did you write a blog? Oh, you did for a virology blog, didn't you?
I did.
Oh, I should
thought it
bring
was
that
in
up.
August. I thought it was in August.
I'm going to look it up. Hang on.
I didn't
Virology
thought it was in August.
blog.
But I could be
What's your
wrong.
last name?
Rosenberg, Rosenfeld, Rosenpenis, Rosenkrantz, whatever.
Yeah, it's January 13th, 2022. Let me do the screen share.
Oh,
This is Test Prudently.
okay.
Let's see where it says about what is a pandemic, though. Does it say anything about pandemic here? The pandemic ends when more of us get vaccinated, when society accepts the fact, blah, blah, blah. I don't know where the pandemic part goes.
Yeah, a definition of pandemic is not the number of deaths, economic burden, but the number of infections. Consequently, it is not correct to state that rhinoviruses are not etiologic agents of global pandemics.
All right. So Raz wants you to explain because Raz doesn't get that.
So you have to. So the problem is what is the definition of a pandemic?
What did Taubenberger say?
So Taubenberger, it says it's a new virus. It's a new immunological virus.
Right.
It has some naive we're immunologically naive, which is also incorrect because by that definition, then every year we have pandemics of flu because there's a variant of flu that we're immunologically naive against and various other viruses. So that's not correct.
I have a definition. A pandemic is a shitorama.
That is true. It is more than one shit show.
Can we put that in the textbooks? Probably not.
I don't know. You'll have to ask Jane. She's in charge.
She won't allow a curse word in Principles of Virology.
You have to ask Jane. So if it's the number of deaths, then that's fine. But there's a lot of things like opioids and cancer are greater pandemics because more people die of them than viral infection. If it's the economic burden, then enters and rhinoviruses are pandemics because not only do you have huge numbers of infections, but the economic burden of people missing school and missing work and stuff is on the par of like $600 billion. So it outweighs this by huge amounts of money. So how do you define a pandemic? So you can't use a virology. Pandemic is not a virology term. It's not a biology term. It is a sociology term. And that
I agree.
Then you have to talk to people about how they want to define it in a society. And then if we're going to define it in a society, then that's fine, but you have to be very clear about what it is. It's not a science term.
It's basically the WHO saying we have a shitorama going on. You need to do stuff.
Yes, basically.
Respond. Okay.
Right. But they have different degrees of shitorama. Because first it goes through, it's the shitorama of concern, right?
Yeah.
Then it goes through, it's the shitorama of emergency, right?
Yeah.
And then finally, maybe 22 years later, they come up with, well, if it's been a shitorama of emergency, then it should be a pandemic.
The important thing you said to take away from this is it's not a virological definition. Okay?
No, it's not.
It's not. Okay, so let's leave it at that. Montreal, Canada. XBB.1.5, increased capability to infect cell types with even lower levels of ACE2, the increased tropism and pos. This is BS. All of this is BS. Has nothing to do with what the virus is doing in people. These are cell culture experiments. We have no idea what the David Ho paper I showed said the ACE2 spike affinity for XBB.1.5 is the same. And so this paper is saying it's different. So it depends what cell you use.
Unfortunately, she's not really a virologist, and she's definitely not a biophysicist. So apparently her kinetic assays are not so good.
And then concern for long COVID. Long COVID concern is absurd to even posit that there would be a concern when you have no idea with these. So no, Montreal, we don't buy this at all. We don't buy it at all.
It just gets her more publicity because she hasn't been in the news for a few months. She doesn't know what to do with herself.
Is this virus behaving the way you would expect it to? I don't know what we would expect because it's a new virus and we were all wrong about, except Amy.
It's not true. I've been wrong.
So I don't think that you can expect anything from any virus.
I think the problem is we didn't really study the models appropriately because we decided that they were unimportant, and then we decided not to give people like Susan and Dennison money to do their experiments to figure things out.
Sandra got Evusheld for our trip to Africa. Is it still effective for killer T cells? No, Evusheld is the antibody cocktail, right? Which is no longer what?
That's what I thought. I thought it was a monoclonal antibody.
Yeah,
I don't know what it has to do with T cells.
Long lasting, you give before you get infected, as they said, they got prophylactically. But it's no longer effective against the Omicron derivative, so shouldn't be used anymore.
I didn't think it was I didn't think you could buy it off the shelf. Can you?
What do you mean buy it off the shelf?
Like, can a physician write a prescription while a pharmacy has it on its shelf?
Daniel's complaining because people are still using it. He says, yeah, we have it in stock. Let's just use it up. It's like useless.
Really?
Yeah.
What is this, milk? We have the milk in stock before it expires. We should drink it? Okay.
Do you know a good animation website that can help students understand how viruses attach to receptors and enter cells? I don't know of any. Maybe some of the listeners here know of some. I certainly don't. We have 481 people, Amy. It's good.
It's good.
It's good.
What's the problem?
How many likes? No, it's good. 218 likes. Hit the like button.
Well, where are the
folks.
other 400? Where are the other 260 people?
Yeah, please hit the like. It helps us to increase our audience. There's a little button somewhere below the video. Yes, John says, make sure your like button is working. Give me a birthday like. Apparently, I actually did leave my 7th decade. A number of people are concerned that, but I'm not changing it. I don't want to leave my 7th decade. I want to leave my 6th decade. Okay. Make me feel better.
So basically, you're going to be the Joan Rivers of virology.
What is she? She keeps going and going.
Well, now she's dead, but I think, but before
she's dead.
Yeah, she died years ago.
Is Joan Rivers dead? Yeah, September 2014.
She died almost 10 years ago.
She was the funniest person. She's amazing, right? Isn't she? Don't you think she's funny?
I do think she's funny. I thought she was funnier. Because she liked to be like 30. She liked to be like 59 for like forever.
So what did she do to be young? I'll try and emulate that.
Oh, no, you don't want to do that.
She died at 94, right?
I don't know. You have that bit right there. Do the math. I don't know what it says.
Joan Rivers died in 2014. I don't know how old she was. Here, Wikipedia will tell us. Well, she went to Barnard. Did you know that?
Yeah, she was very smart.
My gosh, she was born in 1933. She died at 81. That's not very good. I don't want to die at 81, folks.
You know why she died at 81? She had too many facelifts.
Why would a facelift kill you?
All the elective surgery is not good. It's just not good.
Anyway, folks, help Rima with his animations. I don't know of any.
Just not good.
Peter, can you tell me if you received the fourth vaccine? I did not. Amy and I both had 3, 2 originals and a booster. And Amy still has not contracted COVID. It's amazing because she's all over the world. Well, all over Chevy Chase, right? And
I'm in my little vehicle. And
I
my
did
little vehicle
contract it, but
not.
I got Paxlovid because Daniel Griffin told me to do it. Although I'm not an elderly. I'm very healthy, and I really probably didn't need it. But why take a chance, right?
That's what Daniel said.
That's what he said.
And I'm in my little vehicle when I'm all over Chevy Chase.
Your little vehicle, yeah.
Yeah,
You don't like your car,
vehicle.
right?
I hate my car.
Well, you don't like cars in general. You have what do you have, a Prius?
Yeah, I have a Prius.
You don't like it?
So I don't like cars.
Okay,
it's not
in general,
offensive.
it's not the Prius. Okay.
No, it's fine. But like, I went to the Trader Joe's in Georgetown, which is like 5 miles away. So I walked, and then when I came home with my groceries, I had to wait 40 minutes for the bus. And I was telling my mother, and she's like, "Well, why didn't you just use the car, Amy? That's what it's for."
No,
like,
no. You
I
don't
hate
drive
the
to
car.
Georgetown. That's ridiculous.
She's like, "Only my daughter would hate the car." I was like, "Well, tough luck."
So Zachary Foster says VXBB results coming in. Caitlin Rivers is stating that hospitalizations are uncoupled, less people getting sick each day. Says doomsday rhetoric from some people who know better is beyond parody. That's absolutely right. The doomsday rhetoric is absurd. And we saw from Polio Pete how far down cases are now and how far down severe disease and death is. So this is ridiculous. They need news. Okay, so here's our first super chat. Thank you, SRR. You got your stickers. Everybody's happy they got their stickers. That's great. I had sent stickers all over the world, and on Instagram, a young lady in Israel said she got them.
Great.
Works. $1.40. It's so cool. It's only $1.40 to send a letter anywhere in the world. Did you know that? $1.40. $1.40.
I haven't sent a letter. I know, but I'm thinking when I last sent a letter.
Okay, so Patricia liked us starting the live stream with papers. We're going to do that all the time. Okay, cool. Elise says, is there any real way to tell if you have RSV versus the flu apart from a test? No, it's a test is the way to do it, right?
Yeah,
you have respiratory symptoms. You go in. They'll probably first give you a flu test if it's negative. I don't know. Is there an RSV test?
Of course, it's part of the BioFire respiratory panel.
Yeah.
Just as influenza is. I don't know that they give you a separate flu test, and they just don't put it through the bio. That's the whole point in the BioFire repository test.
And Tom likes that I said this is actually quite good, interesting. Yeah, that one paper was the excess mortality. Yeah, it's good. With respect to health and aging, one of the best ways to improve health is to resistance train and weight train. Well, I've been told that cardio is really what I need, but I do need to keep strength up, right, so I don't shrivel away. So I'm going to do resistance training also. All right. Now, I've been doing treadmill for quite a while. I really like it.
Quite a while? You've been doing it for 2.5 months. My God.
Yeah, that's quite a while. What do? You do a stepper, right?
Yeah, sometimes, yeah. when I can't run outside. Yeah.
Reverend Sasquatch, the Paxlovid paranoia is ridiculous. It's a great drug. I didn't hesitate to get it. But if oral REM is available, I might give that a try. Okie dokie.
Well, hopefully you don't need to. Hopefully you won't get sick again.
I remember at the rollout of Paxlovid, you had some concerns. It was just one drug, and the word you used was, I can't remember. We were worried about resistance emerging. I was anyway.
Yes,
probably Amy wasn't.
No, I was too. And then we talked to the guy at Emory.
This is very funny. AJ says, if your head is a mess, I have no hope. So AJ's head must be really messed up, right? That's funny.
I don't know. But, inside, nobody should be inside my head. Even I don't want to go there. And then everybody says, your hair is in chaos. So apparently my head is just a mess.
Oh, I got a letter from someone who said Amy's hair is usually in chaos. I thought that was very funny. You don't mind that, right? You don't take it offensively, right?
No, it's fine.
Would you say it's in chaos now?
Of course.
I've seen you with it properly done, though. Like you go on an interview or give a talk, you have it
Of course.
properly fixed, whatever.
When
right?
I go out to dinner or when I go to synagogue, yeah.
Yeah. All right, here you
Every
go.
Every day? Are you crazy? I get stressed out a minute to it
polio
being at work.
Polio update, Amy.
Okay. Polio update.
What's going on with polio? No more cases, right, of paralysis in the U.S.
Well, no more cases of paralysis have been. No additional cases of paralysis have been observed. I believe that most testing has been stopped in the New York area because they had stopped detecting RNA in the middle of November.
Mm-hmm.
And they decided that after 4 weeks of no detection, it meant that the outbreak was over. I don't think that's the right conclusion because this is not enterovirus. It is not the traditional enterovirus season. So it always goes away in the winter and then it comes back in the late summer and early fall. So that's what's happening.
Okay. MK says, I suspect some of the excess deaths could be people who couldn't get timely care for other conditions due to hospitals being swamped. Yeah, for sure.
For sure.
And Jack has this interesting paper, Why the Evolution of Vaccine Resistance is Less of a Concern Than the Evolution of Drug Resistance. I haven't seen this. Have you?
No.
Maybe we could add this to our stickies, folks. Could you add that? I want to look at this. I want to see why they think. I mean, I would agree. It shouldn't be vaccine resistance, right? Because it's immunity, immunization.
I don't think that it's less of a concern. I don't know what less of a concern means.
Well, we'll have to read the paper. Maybe we'll snippetize it next week.
Okay. Snippetize it? Is that a word?
I can make it up, can't I?
Sure.
Do I have the Amy seal of approval?
Of course. Make up any word you want.
Shitarama is a made-up word, right?
I bet you it is, but no, it's not.
Oral remdesivir
and neither is shit show.
NSP5. No, NSP5 is the protease, right?
NSP3 is the protease.
What's NSP5?
It's an accessory protein.
For what? The protease?
The polymerase, I think. No, for the polymerase.
No, NSP5 is a cysteine protease.
Oh, I was wrong. I thought that was NSP3.
Anyway, it's not the target of remdesivir. Remdesivir inhibits the RNA polymerase. What's the NSP number for the RNA polymerase? Do you know offhand?
14. 10.
NSP12.
Oh, okay.
NSP12. You could keep giving out numbers. Eventually, you'd hit it, right?
But look at it. Look, I guessed right around it on both sides. I was two NSPs off.
Your envelope was not autographed? Okay. Send me another email. I'll send you an autographed envelope. I'll put some stickers in it. Okay? Got some stickers left. Vincent at microbe.tv. It's really not a problem because I have stickers, I have envelopes, I have stamps, everything, and I'm still sending them out. So I'll sign your envelope. I'm sorry, I must have forgotten to sign a few.
Maybe it was before you were autographing.
And I just mailed a batch last week, so maybe you will get yours, Marco. Just keep checking. If anyone doesn't get it within a couple of weeks, Vincent at microbe.tv, I'll send you another one. It's no big deal. Is there any study about what percentage of the global population never had COVID and never got a vaccine? You want global, huh? We could probably get that from one of these websites, right?
I would think the WHO would have an estimate, but I'm not sure how good it is.
These worldometers, right? COVID data tracker of the CDC. Do we want to know that?
No.
Daily update. Raphael, I'm sure there are, and maybe someone can find them as we're talking here. We can go look at it. I'm sure there are, and I just can't locate it.
I'm sure
right
it's
now.
at the WHO.
Oh, thank you, Sandra, so much. Happy 39th birthday. Amy, do I look 39?
Yes.
Come on. Flattery is good, but you can't lie. So if you said I look 50, that's closer to reality. Do I look 50?
Yeah, for sure.
Really?
Yeah.
I don't look in my 40s, though. I don't look 70?
No.
If everyone here said I didn't look 70, I'd be the happiest man alive, like Lou Gehrig said.
Yeah, it's true.
Because I got the great beard, though. It makes me look older. But
no, it doesn't. There's gray beards on young people. It makes you look distinguished, and it's attractive. The gray beard is
folks,
good.
I don't dye my hair, okay? Some people say I dye. I don't. This is natural Racaniello brown hair. Bobby says, we talked about brain shrinkage in mild cases. I asked what could cause this. And Amy said it could be hypoxia, which was great. Has there been any more research into this at all? I haven't seen any. I have not seen any. No. You should just have a Venmo at Microbe TV. Yeah. I should put the QR at the website, right? Yeah.
Yeah, for sure. Have Kara do it.
Okay, so people are saying 50, 55, 59, 60, no more than 60. So I'm happy now.
Okay.
All is good. I definitely don't want to look 70. Oh, my gosh.
Well, you don't.
I would go have a Joan Rivers.
You can't have a Joan Rivers.
Well, first of all, I would shave so I could have no gray beard. And I would dye my hair so I wouldn't look 70. But I don't look 70, so it's good. And Les, can you add to the stickies? Can you add to the stickies to put a Venmo at Microbe TV for the contributions? That would be great. Depleted Uranium Cowboy, thank you very much for your contribution to science communication. I didn't have any cake. No cake.
What'd you have?
We had ramen. We took out.
There was no dessert?
No dessert. Amy brought. Not Amy. My daughter brought ramen. Have you booked the Back to Science lady? Not yet. It's on my list, right, Amy? You sent me her name.
Yeah, probably.
The one she's been on Dan Wilson's page program. Yeah. Everyone is broke after the holiday. Yeah. We had a good fundraising last year, but we have to keep going. That's how fundraising is. You have a good year, then the next year has to be slightly better. So we'll do it. Richard, thank you so much for your contribution. The chat, the super chat works. Thanks a lot. Amy's paper list, they're really bad to the really good. Just because it's there, not necessarily an endorsement, right, Amy? You're just putting papers there that Or
It's not
what?
an endorsement.
What would you call
Some
it?
of them are so poor that I am just stunned that somebody either thought to write it or somebody, you know, accepted it.
Vaxman, thank you for your contribution. Best thing I had after COVID was a salt bagel.
Okay. I used to like salt bagels when I was a kid.
Thank you, John, for your contribution. Zoe says, at the start of the pandemic, experts said masks don't work. Then they said masks do work, but we have to save them for the doctors. Then mask use became mandatory. Why did experts say masks don't work at the start? Amy.
I don't know. I didn't say it.
Because they were wrong. And I don't think that was part of wanting to conserve them for doctors. They were just wrong. They didn't look at the right studies.
It's not even that they needed a study. They just didn't use any common sense logic.
Right. People wear masks for purposes, right? And so we should have been
Yeah,
wearing them to
exactly.
prevent this respiratory infection. So
Exactly.
It's crazy, totally crazy. And that was one of the screw-ups, one of the shit shows that's part of the shitorama that this pandemic was, right, Amy?
Was?
Still is.
Exactly.
Do you have any information on research into treating long COVID with N-acetylcysteine?
No.
I think someone asked Daniel this last week, and he said there really isn't any good trial of it. So no, we don't have any. Just anecdotal stuff, which is not useful, right? Angry Penguin. Polio Pete, Zeke Emmanuel said vaccines don't work on XBB.1.5. Well, that's not right.
Zeke Emmanuel should stay in his lane.
Zeke Emmanuel doesn't know what he's talking about, because what he's saying is the antibodies induced by the vaccine don't neutralize XBB1, which is true. But
in vitro,
in a cell culture dish in a lab. But
right.
Still
in people, that's not true.
We're still seeing protection
Unfortunately, I don't believe that Zeke Emmanuel is an ID doc. I think he might be an oncologist. He should stay in his own lane and stop trying to give advice on everything, because he's just screwing it up.
The key is to see how these variants do with respect to severe disease. And so far, I'm not seeing any uptick in severe disease. It's the same as it was before. So I don't think these neutralization tests are telling you anything about vaccines working or not. Yeah, you're going to get infected. You got infected with Omicron. It was already immune evasive. But disease increase, no.
Okay, so
wait.
What does an oncologist bioethicist, who is the chair of the Department of Medical Ethics, know about ID?
He doesn't. He's out of his lane, and the press go after him, and he says this stuff, and then people get scared, right? Vaccines don't seem to work.
They go after him because he's Rahm Emanuel's brother. I think they're twin brothers. Oh, yeah? If I remember correctly.
Why don't they go after me? Why don't they ask me? Or you even? No, you can't, because you work for the government. They could ask me, but they don't. They don't.
Well, they should, but I don't want them to ask me. I'm too busy.
Actually, I was asked to go on China TV tonight.
So?
Well, I couldn't, because we have this stream. This has precedence. I've been on it many times before.
They couldn't reschedule?
No, they wanted to talk about the situation in China. You know, an outsider like me, I'm going to talk about the situation.
I was going to say, when are you going to China?
I'm not.
You could dig to China.
What did you say?
You could dig to China. That's what people say when you dig big holes in the sand at the beach. Or you dig
you could dig to China. You could dig. I couldn't understand what you were saying. Oh, my gosh. Okay. Ian says, is selection a passive consequence? Yeah, it's passive. So as Amy said earlier, viruses replicate. They throw off all kinds of mutants. Among them are some amino acid changes that confer resistance to an antibody. And if that gets into a person with the antibody, that virus will predominate. It's a chance occurrence. Yep. Is that fair, Amy?
Yeah.
David thanks you for your thorough and thought-provoking answer to his question regarding cell culture testing versus clinical monoclonal efficacy. I think that was last week, right? Or maybe it was today.
I don't know. But you're welcome. I'm happy to help.
Thank you, Karen. Thank you for your contribution. Really appreciate it. Will SARS-CoV-2 ever stop mutating? No. No. Never.
Yeah, when the polymerase dies.
When there's no more SARS-CoV-2. Is that going to happen ever? No.
No.
No. Every time it reproduces, every cycle, which means it gets into a cell, more viruses are made, it's mutating. And Andrew makes a good point. He eats viruses every day, mostly plant viruses. We all do. We have lots. The most abundant viral RNA in our feces is a plant viral RNA, mild pepper mottle virus. Did you know that, Amy?
I've taken your virology course several times, yes.
She didn't ask if it was blunted. Amy, classic line. Yes, right. That's what you said to me like 45 minutes ago. It is a classic line. Unless you're very specific, do you make an immune response? Amy says, yes. And Vincent says, but it's blunted. And Amy says, she didn't ask if it was blunted. It's very funny,
true.
I have to say. It's very funny.
I should be a comedian.
If a first-rate cardiologist decided not to recommend COVID therapeutics in a high-risk individual, would you question their expertise in cardiology? I don't understand what the cardiologist is. So the cardiologist is consulted because the patient has a heart issue, right? Well, if they think that
I have no idea. I don't understand. I don't really understand.
What we are questioning are cardiologists and oncologists talking about vaccines and viral infections because it's not their expertise, right? And
yeah,
so
because it gets
Emmanuel,
it gets them in the press and it gets them visible.
And Emmanuel, well, I don't even think they care to some extent. They just get asked the questions, they give the wrong answer, and it propagates. Because Emmanuel, it's an MD. People think every MD on the planet knows everything. I don't know anything beyond viruses. And that's obvious when I try to tackle, like, shoes. I don't know anything about shoes.
I only know a little bit about what very specific virus.
My father was a surgeon. He knew Zippo about viruses. I mean, he died 40 years ago.
But he knew a lot about the heart.
Yeah, he was a heart surgeon. He knew a lot about the heart. But if he had to deal with infections, he'd call in an infectious disease physician.
Right, but he was really good with the heart.
He was a good surgeon, yeah. That's why my hands are very steady. See, no shake. I got genetic steadiness.
Good.
Is it possible for the estimate infection fatality rate of triple vax person in 2022 to increase in 2023, the future?
I don't understand the question.
Well, you want to know if triple vax people, their infection fatality rate increases. You could estimate that, I think. Why not? I'm not sure anybody would, though. Right? That's got to be a careful, expensive study.
But as more and more of the population is triple vaxxed, then more and more of the people who die are going to be triple vaxxed.
That's correct.
So I don't understand.
By the way, we have 511 people. It's good. Without Daniel Griffin, Amy.
Yeah, I'm more concerned. Where's my 300 people with their like button?
Please hit the likes. Should you take Paxlovid even if you don't feel too sick, if you're at risk in a risk group?
Sure.
Well,
go ahead.
Well, you have to listen to what your doctor tells you, right? That's how you're going to have a prescription. So, No, I wouldn't say sure. If you're not in a risk group and your physician doesn't prescribe it, then you don't take it.
They are in a risk group. They just don't think that they have a lot of symptoms.
Oh, if you're in a risk group, yes, you don't wait to have worse symptoms. That's for sure. Yeah. How many shots of the Chinese vaccine would equal 3 shots of Pfizer or Moderna? Well, if it's a poor antigen, it doesn't matter how many shots you give. It's never going to come up to snuff, right? It's not going to work. It's not going to work. Thanks, Kip and Laura, for your contributions. Really appreciate it.
And Emma.
Yeah, somebody wanted to visit the incubator today, but they didn't come.
Oh. Sorry.
Thank you for your contribution, Mr. Seaman, to science communication throughout 2023. Okay. Do you have a recommendation for a source that describes the process that separates the genetic material for each source in a sample containing material from a large number of viruses? You can't separate it, but you can do high throughput sequencing and get the sequences of all the different genomes, right, Amy?
Yeah, you barcode them.
You barcode them, and you'd have to do bioinformatics afterwards to assemble them, correct?
Yes.
So let's explain this a little bit. If anything, for me, let's say you get a soil sample where you think there's a lot of viruses in there. You extract the nucleic acid, and then you convert it to DNA.
So using primers, you can use primers for RT.
Yes, reverse transcription. That have a barcode.
That have a barcode.
Okay.
And then you amplify that with primers that are complementary to the barcode, and you could have a second barcode in the DNA primers. Yeah.
So a barcode is simply a primer that you know the sequence of, so that later you could.
Well, it has a barcode is you put 8 nucleotides at the 5' end that doesn't anneal to anything of a known sequence, and then you sort by that known sequence. So like you can have 8 A's and 7 A's and a G and 7 A's and a T and blah, blah, blah. Different combinations. Like the barcode on the bottom of your cereal box.
Got it. And so you don't actually separate the nucleic acids, but you can get the separate sequences, right?
Right.
So where could you get a source of this? Go to the Illumina site.
Watch the Illumina video at the Illumina site.
Yeah.
They have a movie.
How do you spell Illumina? ILLU?
ILUMINIA. Illumina.
Okay. I remember Dr. Griffin saying Paxlovid reduces hospitalization from 1 to 30 to 1 to 200 in the elderly. Accurate? I don't recall. Do you recall?
I don't know.
I have to go back to the episode, but Daniel would know. I could text him right now. Let's text him. What do you think?
Okay, go ahead.
Here, let's do this. Daniel. Where's Daniel? Just give me a second. Daniel Griffin. Does Paxlovid reduce, what was the question?
Hospitalizations.
Oops.
In the elderly.
In elderly from what to what?
From 1 to 30 to 1 to 200.
Okay. Bingo. Let's see. It's 9 p.m. You should be home. If someone wants to know if you're okay, they think your voice is low. Are you all right?
Fine.
Okay. Elise says, do we obsess about neutralizing antibodies with any other vaccine? Nearly all the news has been about the wrong thing. It's true. No, we don't. Antibodies neutralizing flu antibodies are never in the news, right? Polio neutralizing antibodies are never in the news. Norovirus, any virus.
Well, we don't have a vaccine against norovirus.
True. So it's just the extent of the pandemic has done this, Elise, and there's too much press and too much of the wrong press. And you guys who come
They
here,
must be bored. They don't have anything else to write about.
and you guys who come here get the lowdown. We've been doing this for 2 years now. They're getting the lowdown. You're getting what's right, okay? And from the very beginning, I said it's not more transmissible, and it's not. And everyone said I was wrong. Vincent, you don't know what you're talking about. No, I wasn't wrong. It's basic virology, folks, not epidemiology. Basic virology, it's immune evasion. Okay. Apart from, I just had to rant a little, Amy. I hope you
It's fine.
forgive me.
I don't care.
Apart from funding, what are the factors that can make study of viruses more difficult for lab researchers? Is there any current interruption in supply chains? Well, Amy will tell you about supply chains. When she was at Columbia, she'd order serum, and how long would it take? 6 months to get it?
Oh, longer sometimes.
Which normally would be a few weeks, right?
Couple of days.
Plasticware was horrible. Okay, all of the supply chain was forcocked.
It's still
during
not
the
so good.
Pandemic.
It's still not so good.
Funding, funding. What you say apart from funding, people. You need people to do the experiments. And
You need people, but there's a lot of viruses like norovirus that don't really have culture systems.
Mm-hmm. And they're also dangerous. You need a BSL-4, or with SARS-CoV-2, you needed a BSL-3, and that stopped a lot of people from using infectious virus, and that's why we're stuck on this goddamn PCR assaying everything, like that paper looking at nucleic acid in tissues. They should have been looking at infectious virus, but they don't have a BSL-3.
It's because it's easy. It's because
easy way
it's
out.
easier. It's the easy way out, and people make stupid assumptions like that virus equals RNA. And the fact is they probably walked down the hall to their local virologist, who then employed them with the wrong information because they probably were poorly trained. It's the way it works. To get trained properly is a huge investment. You should be happy I appreciated it.
It took me 20 years to train Amy. I'm
22.
just kidding.
22. Took 22. Get it right.
Does the Paxlovid hospitalization reduction apply to the vaccinated as well? It does apply. I don't know if the numbers are exactly the same, but it does apply. Yeah, absolutely. No, it's not my birthday today. My birthday was the 2nd, so Monday. It's okay. But I would definitely do this on my birthday. Yeah, no problem. Do you think being vaccinated and previously infected makes you less likely to shed virus during a subsequent infection as compared to someone who's never been vaccinated?
Yes.
Yeah, for sure. I saw a paper today, Amy. It's one of your papers in the Amy papers saying that vaccinated people clearly shed less virus.
Was that by Raul? That might have been by Raul.
I don't remember.
He said he came out with a paper similar to that, I believe.
0 to 9 is the first decade. I didn't know that.
Yeah.
Sorry, I left my 6th decade. I'm not going to my 7th. I'm changing the number.
Fine. Move on.
The Joan Rivers of virology. You like the pink sculpture on the mantle. Yeah, so the pink sculpture right there, that is a Luke Jerram glass sculpture. I'm not going to go get it because I don't think it would be a good idea. It is gorgeous. It was given to me by my microbe TV colleagues on the occasion of the 600th TWIV. Now we're going to hit 1,000. What are they going to do for me then? Anyway, it's really beautiful. It's sitting on a little light.
I'm renting you an entire theater to have a big thingy. My God, what more do you need? Your name and lights?
No, I don't need my name and lights. Amy found a theater in New York that'll seat 52 people. We're going to try and get it for TWIV 1,000. So if you're in the New York area, it's going to be sometime in April, probably a Saturday. We're going to book this theater, and 50 people can come. Right, Amy?
They have to make it's first come, first serve, and by donations. Yeah. Donate. Mm-hmm.
Although the first 2 doses of the Chinese vaccines are less effective, A third dose brings them up to similar levels of protection to the mRNA vaccines. And so many people don't have that third dose. That's a problem. But they also have a lower vaccination rate, right? That's also a problem. Amy, how unhealthy is it to eat food that has some mold on it?
You just cut the mold off of cheese.
And it depends on the mold. Some molds are worse than others, right? So I cut it off, and I think it's fine. Cheese, bread, I don't have a problem with that. And I'm still here. I've left my 6th decade of life, and I cut the mold off my food.
I think you're fine.
Sam says everybody and their dog can measure antibody neutralization and get attention from New York Times WAPO reporters. It's true.
That's true.
I think you had someone on data 2 years ago saying flu mutated 7 times faster. Mutated faster than what? I don't remember that.
I don't know.
Sorry. John, thank you for your contribution. Since I don't have a question, I'll remind everyone about Smile Amazon, which can put a portion of your purchases towards Microbe TV. Yeah, smile.amazon.com. You designate Microbe TV as your charity. It's in their list. And then whenever you shop at Amazon, you have to go to smile.amazon.com. If you go to amazon.com, it doesn't work. So that's kind of a barrier, right? Amy, do you go to smile.amazon.com? You don't use Amazon. You do? Did you designate Microbe TV as your charity?
Yes, when I use Amazon, yes. I don't buy that much at Amazon. I'm one person. How much do I need?
Yeah, blue cheese is
Can't
made
have
with
a
mold.
whole house full of crap, you know.
It depends on the food and the mold. That's absolutely right. Blue cheese has mold in it. Many cheeses have mold naturally, but you have to be careful, yeah. All right. COVID-19 virus found in the brain, December 14th, Nature, to isolate virus in cell culture from multiple non-respiratory tissues, including the brain. The problem was they didn't quantify it. They just threw it on cells and it grew out, so we don't know if it was 1 PFU, which would be nothing, or more. So everything is flawed. I always substitute more transmitted than for more transmissible. That's a good idea, actually. It is more transmitted because of the immune evasion. Yeah, very good. My 9-year-old has just finished a 10-day course of antibiotics for strep A. Is he more susceptible to enterovirus infection in the short term?
No.
The eyes open wide. You can see the whites of your eyes.
I had to do some thinking.
Yeah, I think it's fine. No, there's no evidence that antibiotics is going to affect enteroviruses in your gut.
Or even in your respiratory tract.
Elise says, my 25-year-old won't get another booster. He says 3 shots are enough. I think he's probably right. Should I still nag?
No.
Your 25-year-old is listening to us. No, you don't have to nag. That's enough for a 25-year-old. Yeah, no problem. This listener, Amy, wants to hear you and Michael Schmidt in a live stream. Michael Schmidt would be what's a word for a live wire?
He's a live wire. I don't know.
A pip, right? A pip.
Yeah, he does yeah.
All right, here's a good one. Maybe a norovirus pandemic would be a shitorama.
It is. It is.
It is a shitorama. 4, a 40-year-old healthy person with no infections or vaccinations. How many mRNAs would be good enough? 2 shots 6 months apart? I think 2 shots 6 months would be great, but you're supposed to do 2 close and then the booster farther apart. That's the official recommendation. But, Amy, don't we think 2 shots 6 months apart would be great? That would be sufficient diversification of the antibody response. But even Paul Offit would say you need 3 to get the best breath.
Well, if you follow the original regimen, probably, yes.
Yeah, so the original was 2 shots, 3 weeks apart initially, right? 3 to 4 weeks.
Mm-hmm.
And then the booster was months down the road. So I think 2 months, 6 months apart for 2 would have done the same thing, but nobody ever checked that, as far as I know.
Yep.
Be careful with swear words because YouTube will demonetize you guys. Well, we don't monetize. Okay? I don't monetize anything. You guys give money here. They can't do anything about that. Well, they could take it away, I guess, but the SHIT is a mild word. It's actually a medical term, right, Amy?
Yes.
If new subvariants evade immunity better, will a booster help with better protection? No, it won't. For a few months, you'll have a boost in antibodies, but then after that, you won't. It will go down, and you'll get infected, and you'll have the same issue. Plus, the variants keep changing, right? So you get infected, you get mild disease, but you're protected from severe disease and death. And, people say, well, I don't want to get any disease at all. And Paul Offit says, we don't have vaccines to prevent mild disease. That's not what they're about. They prevent severe disease and death. We don't have, for example, Amy, a vaccine to prevent subclinical polio, right? Subclinical paralysis.
I'm sorry, what? I wasn't, my earphones were not working properly. I thought I heard the word subclinical paralysis.
I'm joking.
I know. It's an inside joke. I understand.
I had the flu vaccine a couple of months ago. How effective has it been this winter? Oh, I saw some of the first reports on this, right? I don't know. Do you know, Amy?
I don't know. I don't keep track.
I know there's a lot of flu. Of course, everybody knows that.
There's more flu than SARS-CoV-2.
Match, according to the director of the CDC, which would be
Michelle.
Yep. What do we have here? 539 and 413 likes. That's not bad, actually. It's probably the best that we're going to ever do, right?
Why? You're giving up? You don't think you can do better? That's not like you. You can do better. Push the button.
Do better. Let's push the button.
Push
get
the
it
button.
Up to 4 something. It's 537 people. Let's get it over 500. That would be a challenge. All right.
Everybody should be up to a challenge.
Pete says there was an animation of SARS-CoV-2 spikes springing in cell entry, a pic in TWIV in the last few years or so. That would be okay. That would be okay.
Sure.
Yes. Joan Rivers is dead. I thought one of your lectures had an animation showing a virus binding and entering the cell. Yes, actually, we have influenza virus binding and entry. We have HIV binding and entry in my lectures. Those are old innovations. So, Rima, here we go. There's a company called Ex Vivo, and they do animations. And look them up. Ex Vivo. Yeah, thank you. So, Jeff, you jogged my memory. Yeah. So apparently, Joan Rivers died during surgery. Here we go. Liz says,
okay,
doing
not
OP.
so good.
What's OP?
I don't know.
Okay. That's kind of like,
surgery?
didn't Andy Warhol die at Cornell?
Outpatient surgery.
Outpatient.
OP is outpatient surgery. Andy Warhol died from a botched gallbladder surgery, yes.
And Jim Hansen died of
pneumonia. No, I believe he died of pneumonia. Look it up.
Plastic surgery procedure. Okay. Elective surgery. A lot of Joan Rivers here.
I know, you say I know nothing and I'm not hip, but, I know stuff.
I never said you're not hip. I ask you for the hip questions. When I have a question about some art or stuff, I ask you. Okay. So stop it.
I know stuff.
Any differences in release of safety data with mRNA vaccines versus others? No, not at all. And I mean, I know people are making a big deal out of this, but I see no problems whatsoever. No problems. Janet Iwasa, yes, that she was another animator. I think she has some innovations too, Rima. Janet Iwasa. There you go. There you go.
Utah.
Jeff, thank you for your contribution to science communication. Wait, is Amy Rosenfeld in D.C. now?
Yes, I is.
You've been there for 6 months?
Yes.
So Amy is living in Chevy Chase. She's working at the FDA, and she has to drive there, and she doesn't like it.
That is true. But I go to D.C. à pied all the time.
Au pied?
On foot.
On foot,
yeah,
au
on
pied.
Foot. Yeah.
Yeah. Midge says, do you think it's okay to have had 2 vaccines, AstraZeneca and 1 mRNA? Do you think I should get another mRNA vaccine? That's a good question.
I think before the recommendation was that you only needed 1.
All right, so Daniel answered the earlier question.
Mm-hmm.
Pre-vaccine, about 1 in 5 hospitalized overall, worse in the elderly. With vaccine down to 1 in 50 overall, but worse in the elderly. So 1 in 30 is a fair estimate for the elderly, but Paxlovid only about a 60 to 70% reduction in the vaccinated. So that gets me to about 1 in 70, not 1 in 200. As one is probably trying to apply the 89% reduction in the unvaccinated. Okay, folks? There you go. Thank you, Daniel. So did we decide that Midge is okay?
Yes, I believe so.
Ben, thank you for your contribution. Is it true that Africa, besides South America, is largely untouched by COVID? South
I don't
America
think that's
is
correct.
Largely untouched. No,
I don't think any place is largely untouched by COVID. You have to ask Daniel about his experience in Africa, but I don't believe so.
Let me ask him: Has Africa been untouched by COVID? He's probably going to say, "What are these from the live stream? Why don't you get me back on?" But South America, we talked with Esper Callis about how Brazil got hammered, right? Of course, it's only one country, but
it's quite a large country.
It is. Oh, Midge put her sticker on her car. That's cool. That's very cool. Excellent. Thoughts on how China is handling COVID? Not well. So they have low vaccination rates, right? Especially in the elderly. They were locked down for a long time, so they didn't have a lot of population immunity. They decided to come out of lockdown. And I've heard some of the officials saying because Omicron is mild, well, that wasn't a good idea. Now the virus is ripping through a largely non-immune population. So what could they have done differently, Amy? I think they should have vaccinated more people. You know, you think in a country where they can keep people in their homes, that they could vaccinate them? You know, go to the homes and vaccinate people? That's what I would have done. I mean, if you're going to be
authoritarian.
Thank you. You read my mind. Was it ugly up there when you went into the mine? No, it wasn't so bad.
No, it was much better than mine. It's much more organized. Mine is not organized at all. There's shoes and clothes all over the place.
You would think an authoritarian country could vaccinate its population. So I actually don't get that. Maybe someone has some insight on that. Until there is enough supply of the latest cell culture validated therapies, monoclonals, would we not continue to use the washouts? Cell cultures do not tell the whole story. I asked Daniel that last week. I said, hey, maybe these monoclonals actually do work, even if they don't neutralize. But there isn't any evidence for that. It's a good point, right, Amy? There could be FC-mediated functions but you'd have to show that they are clinically useful, and nobody's done that experiment. So no one's done a trial.
I wouldn't know anybody who would have said last week that FC and binding and Fab activity were not mutually exclusive. I don't know who would have said that.
That must have been Mickey Mouse, right?
Oh, I was thinking Goofy. I'm much more of a Goofy dog fan than a mouse fan. The mouse is kind of annoying.
All right, here's a new listener. So welcome, DJ M. Changes to our DNA. Came back was inconclusive, but I'm presuming you're referring to getting an mRNA vaccine. And no, it does not integrate into your DNA. In Cells and Culture, they had some people said yes, it integrated. Other people said no, it didn't. There's no evidence that it integrates into people.
That's because the cancer guy should have stayed in his own lane. He screwed up cancer, and then he decided to screw up this.
So DJM, there's zero evidence that these mRNA vaccines are causing any
transformation,
and I wouldn't worry about it, frankly. Okay. All right. Janet likes your bun. What were you eating a bun before or something, or what?
The hair.
Oh, I didn't see that. If pandemic is not a virology term, then endemic too. Do you think that's not a virology term?
Yes, that's not a virology term either.
And Meister says all words are made up. Well, that's true, but we need to know their meanings when we make them up, right? Otherwise, we're in chaos.
Why we have dictionaries that use the word that you're supposed to define in its own definition.
Don't you think there'd be chaos if we made up words and we didn't know what they meant? Or at least we didn't agree on what they meant? Yes. Hello, Amy. My friend and I will be traveling the U.S., plan to go to Washington, New York, San Francisco. I would love to take you for an intellectual and fun lunch.
Okay, contact Vincent. He'll set it. He'll forward you your information to me.
What am I, your matchmaker?
You're my pimp now.
Matchmaker,
Make me a match.
Make me a match. Yes, Vincent at microbe.tv. I think you already have my email, and then I'll forward it to Amy. And maybe I could visit the incubator. There's nobody named Vince at the incubator, though. Do you know anyone named Vince at the incubator, Amy?
I don't know anyone named Vince in the incubator or out of the incubator. I know of Vincent, but I don't know.
I prefer to go by Vincent. Could iodine spray in the nose and throat make it harder for a virus to gain a foothold? It would be transient, if anything, right? Because it's going to wash away, and then the virus will still be coming in, or if it's reproducing, it's not going to make any difference. So I don't think such treatments are really effective, such spray treatments. The sports world has been swamped by anti-vaxxers claiming this Buffalo player's cardiac arrest was from an mRNA vaccine. Yeah, full tilt BS. You bet.
Yeah, I mean, he was violently hit. No, I'm serious. He was violently hit. He went into, it's all on, live television, him going into cardiac arrest and everything. I mean, it's kind of traumatic to watch.
A hit can do that, right?
Yeah, especially a hit in the chest. Yeah, the force of the hit in the chest. Yeah.
Many years ago when I used to watch sports, when I was a wee thing, I remember there was a quarterback who had his leg shattered on the field.
Yes, that was Washington.
Joe Theismann. Right? Oh, man, you know this stuff. Holy cow. Wow, I'm impressed.
There's also been people who've been paralyzed on the field.
I've just told my T cells I'm watching TWIV. They will get an education. That's very good. It's very good. Love it. Okay, I'm looking 50. Someone says I don't look 39. No, I don't look 70. Yeah, but I want to look. Okay, Raphael, 50, 55 tops. 59, not a day over 57. So on New Jersey Transit, I have a senior card, right, which is 65 and up. And the guy always gives me a hard time. He says, you shouldn't be using this. You're too young. And I said, no, I'm 69. And he says, I don't believe it. So you guys are right. 60-ish. I'm very happy that I look so young.
So when I say this, it means nothing. But when these people say this, it means it's super important.
Hundreds of people are saying it, Amy. It's as opposed to one. And even though you're a big one, it's good to have numbers.
Say,
Look at Geraldine. No, I don't know what the G stands for, but she's framing her envelope that I signed. Isn't that cool?
Cute. Very good.
Yeah, it's the hair that does it. Don't do it, Joan. Researching virology keeps you looking youthful. Yeah, no way you're 70. How do people get stickers?
They write to you.
Write to me, Vincent at microbe.tv. You need to send your postal address, right? And I will mail you in an envelope that's signed on the back, so you could either throw it out or save it. And because maybe in 50 years it'll be worth $5 on that old auction show. What is that called?
Antique Roadshow?
Antique Roadshow. Well, I found this autographed envelope from this virologist, Vincent Racaniello. What do you think it's worth? And the guys, well, this is really worthless. But then they wouldn't bring it up to the front if it were worthless. Oh, sorry. Will you comment?
I've never
iodine
watched
spray.
The show.
No, iodine spray is not going to work. It's going to be transient at best, right? And it's not going to be good for your mucosa either.
It's not a brilliant idea. I don't know who would have suggested it, but not a brilliant idea. Not good. It's just not good. I'm sorry, but not good. Not brilliant, not good.
A lot of age stuff. People really reacted to my age thing. Thank you, everybody.
When I say stuff, nobody cares. When 400 people say, you care. Like chopped liver without a Ritz cracker.
Anyway, this class switch paper is no concern whatsoever. You know, the headlines, the anti-vaxxers are making it a concern. They just don't understand that it doesn't mean anything. Anti-inflammatory is fine, okay? It's crazy. All right.
Yep.
Margot, thank you for your contribution. I notice you're here every week. I recognize some of these icons. Thank you so much for helping us out. Okay. Depleted. Okay, what else? Language. Yeah, I'm sorry about the S-arama. Okay. I won't do it again.
Why? Shitorama is fine. It's a shitorama. I say it all the time. Even though I have other people saying it, and they have the appropriate definition. In order to be a shit show, you must be a minimum of 2 shitoramas.
Okay, let's see. We're at the beginning of the pandemic. It was believed COVID was passed by fomites, and if people were masked, they would keep touching it. Okay, I don't think it was all fomites in the beginning because this is a respiratory virus, right? But someone said that Debunk did a thing on the IgG4. Okay, that's fine. Go look at that. We didn't do that. We'll probably get to it. We'll do the science, yeah. HA protein is the main immunogen in inactivated flu vaccines. The mRNA-based flu vaccine would generate a strong cellular CD4 and CD8 T cells, and we may stop chasing the variants.
I don't think my flu guy will agree with that.
Oh, you have a flu guy over at the FDA?
I have a flu guy.
So
he does not think that. Try again.
T cells are not the key to preventing severe influenza? I thought they were.
I don't think he would say that HA is the main immunogen. I think he's looking. I think that most HA assays are influenced by the presence of NA, so it becomes an NA-HA assay. You have to do an NA. In fact, actually, I think if you did an NA assay, if you look at NA only, NA is actually a better immunogen than HA. And so therefore they should, my flu guy would not agree with that.
Okay. There you go. It's from the horse's mouth.
Now I'm a
Hello.
horse. First I was chopped liver. Now I'm a horse. I'm moving down the food chain. And I didn't even get a Ritz cracker with my chopped liver. This is really not a good night.
Hello, Unstable Unicorn, who I see on Instagram. You signed up. Okay, so send me an email, Vincent@microbe.tv, with your address. I will send you a mug and I will send you stickers. And anyone else who's a patron on Patreon, if you haven't gotten whatever your reward is, just write to me, because in the next few weeks I'm going to actually ask everyone, okay, you haven't got what you were promised. Tell me if you want it. The tiers are mugs and T-shirts. So just send me an email, Vincent@microbe.tv, and I'll take care of it.
I thought Kara was going to do all of this.
Well, Kara's going to do it going forward. We're going to change the rewards now to something less onerous, so like a sticker and something else for the higher tiers. But just let me know if you want your reward. I'm happy to send it out. It's not hard. A few people have already told me, and they will go out as well. We dug to China every summer. You must have gone to the beach, right? You remember going to the beach, Amy, and saying we're going to dig to China?
Yeah, I go to the beach every summer. I like the beach.
Did you see the recent video from MedCram, a new infrared light? No. So was Far UV. Remember, that was supposed to be a big thing. We had him on Twiv, but there's been limited deployment of that, so I'm not sure what's going to happen with this. Fun fact, if you dig a hole to anywhere on the globe and jumped in, it would take 42 minutes to reach the other end. 42 minutes at what speed? You would just be falling, Amy? Would you fall into this hole? I guess so.
I don't know. First of all, the pull of gravity is always 9.8 meters squared. So you're accelerating at a certain pace. So,
So,
and
yes, it must be falling in, yeah.
you'd have to fall in, and what is it? D equals AT, and it's always the same pace.
Already missed.
Why would you have a vegan birthday cake?
Hang on, already missed says chopped liver on a Ritz cracker. Love it. I didn't have a vegan birthday cake. I'm not a vegan. My daughter says I'm a pescatarian.
You eat other things besides.
I don't eat meat.
You just don't. You
I
eat cheese and eggs and all the other stuff. You just don't
eat meat. I don't eat red meat. I eat fish and cheese and eggs. Yes, I'm not a vegan. Whatever you want to call it. Just don't call me late to dinner.
Don't worry about it.
A while back, I recall hearing that those who survived COVID then got at least one jab were considered very protected. Did we ever find out if vaccinated then survived was equivalent?
Yes.
Yes. Yep. There have been a number of such studies. Yeah.
Apparently, my physics is not so good. Pete Glass is going to correct it next week.
Monovalent supposedly only 29% after 4 months now. 29% for what? No, this is not right. Ashish and Fauci are wrong here. Okay, the new bivalent boosters do not help in terms of severe disease. 29% is just infection. Yeah, no vaccine is preventing infection after 4 months. Can we get this in our heads? Not you, Brittany, but these spokespeople, Ja and Fauci.
Well, Fauci's retired.
Fauci's gone. Fauci's gone. Yes. Goodbye, Tony. The original vaccine still produces T and B cell immunity against severe disease. Memory. As far as I know, bivalent doesn't work better in this regard. That's right. There have been no studies. They're pushing the bivalent, but there are no studies that say it's better. And I don't think that's right, because then they're going to say we're going to have a new bivalent every 6 months, which is just wrong. So they better do the studies. Okay. I've been seeing more about, mass extinctions of species. Does this mean less zoological virus spillovers? No, because most of the spillovers don't come from all these extincted species, right? They're mainly from rodents and bats and birds. And last I heard, Amy, those were pretty plentiful, right?
Yeah.
They were
I'm
pretty
not insulted
plentiful.
that I didn't factor in rotational effects. The guy thinks I'm insulted. I'm not insulted.
No. Amy knows she doesn't know about rotational effects.
I did a fast, cheap physics.
So, Brittany,
I
the
just said
bivalent booster
everything.
has BA5. The predominant viruses are beyond BA5. So, you can't say that it's going to be better. And anyway, the data don't show that it's better. We talked about this last week. What the CDC released is simply showing that a couple of weeks after the bivalent booster, yeah, you're protected against infection because you have high antibody levels, but that's going to go
They
away after a few
repeated the
days.
baddest. So they repeated the bad Israeli study from 2 years ago. Is that what you're
That's
telling
right.
me?
That's right.
Oh, that's a good plan. It's a good plan. I like that plan.
It is a good plan.
If I repeat bad work like Flexner, can I get publicity too?
You can.
Really? So you think I should go and repeat the bad work that Flexner did for my virus?
No. You know better than that. Steve says, have there been any new studies on how long a person still is after their symptoms end? I wonder if quarantine is still necessary. 7 days, but CDC says 10 days still, right? So
I thought it was 5.
Well, I think 5 should be a good one, but I don't think that's the official CDC recommendation. And this is based on
I don't know.
The variants, but I would say 5. So Daniel says 10 days. 7 days, I think, is really realistic.
I don't know. I don't follow it.
We will all fly in for the 1,000 TWIV. Why don't you auction off tickets? So, Liz, I think
he's going to.
I don't think anybody's going to come. So I figured the room will be empty. Maybe Liz will come. So do you say Liz?
Oh, okay. So
Lise?
let me get let me get this straight.
Wait a minute. Let me finish with the name. Is it Lisa or so Liz or Lise? How would you pronounce it? I don't know how you're going to tell me. You have to tell me when you come for the 1,000. Okay. So we should do some auctioning of tickets.
Yeah, they have to. Yeah, they have to make. Yeah, you auction or you may say a ticket is a donation, but to get your ticket, you donate to Microbe 2EV between $25 and $50.
Well, people have to send in an email of intent, and then we just pick them out of a bucket, and then they have to pay, right?
Yeah.
So we can raise some money, but not an exorbitant amount of money.
You have to cover the stuff. Yeah, it's fine. It's what people do.
Are you thinking you want one day to invite all the Twivers to a Zoom meeting? You mean like all the thousands of listeners? I don't think that would be a good Zoom meeting. But what we're thinking of doing is doing some live stream Twivs. I'm thinking of doing it now and then. Do it. So we record on Fridays at 3 p.m., right? And just put it on this platform here and live stream it on YouTube. And the cool thing about that, Amy, is as soon as the episode's over, the video is done. Yay! I don't have to do any editing. What do you think about that? Look, the video is pretty good here. Mine is spectacular, of course. Yours is, we need to get you a new camera, but we'll do that. I'm going to give you the one from my office that you used to use. That was much better. I'm going to give that to you. So next week, we're going to go clean my office out, right? So I'll give you that camera.
He never responded. I'm going to have to. No, I'll take care of it.
Okay.
Don't understand. I send nice email. I say Happy New Year. I ask nicely. No response. Everything's a problem.
I need a light up Joan Rivers statue to commemorate his upcoming YouTube milestone. What is my YouTube milestone?
I don't know, but we don't need no Joan Rivers statue. No more crap.
I have 116,000 YouTube subscribers. So I'm not aware of us reaching any milestone. Oh, we're at 472 thumbs up. We need less than 30 to hit 500. And
That's
that,
great.
together with the fact that I don't look over 60, that'll make my day. But the day is almost
Make
over,
your
of course.
I say you can no longer complain for a month.
Okay, that's fine. Krista wants to know any new info on the effectiveness of Novavax or value of boosting with it rather than with an mRNA. Not yet. These are just being deployed. So I'm presuming in some months we will have data because we're going through the season now, right? The big season. So we should have that. Yeah. This is very funny. People are talking about molds. The guy
wanted a fungus podcast, right? Rob wanted a fungus podcast. Now he's got one.
People are eating molds. Oh, gosh. Some of these questions I can't answer. Are there other RNA viruses besides coronas that code for proofreading enzymes?
No.
Corona's the only one. The nidovirales, which is the order that includes the coronaviridae, is the only one. Yeah. Oh, here we go. Ron Fouché said flu 7 times mutational drift than COVID-2. But Amy would question that, right?
He would know.
You would know, yeah?
Ron would, yeah, for sure.
Okay. So the USDA says for moldy soft cheese, throw it out, but hard cheese, cut out the moldy part. Okay. Got it. Professor Racaniello, your colleague, Professor Rosenfeld, would love to receive an Italian coffee maker as a gift. I don't know why it's a joke. Do you? Do you have an Italian coffee maker?
No.
You don't want one, right? Yeah, that's probably good enough. Right?
I have the same one you have.
All right. The UK spaced the initial vaccines in the beginning. Wasn't the timing 5 or 6 months? I don't think just 2 doses worked well. I think I do remember that they did, in fact, a study where they compared the spacing. I'd have to go back to that and look because I don't remember. Take a look at that, Elizabeth. Yeah.
3 more minutes. People should push the button.
Environmental, thank you for your contribution. Yeah, but by monetized, I mean turn on the monetized button right when you post a video. I never do that. That's what I'm talking about. Yes, I understand this is monetized, but in a different way. And I don't care if YouTube says it's the same way. For me, it's different. I would guess that one reason vaccines can't protect against mild illness is due to the immune response. Yeah, but there's virus reproduction. That's the problem is if you have memory T and B cells, it takes a few days for them to kick in. And in those few days, the virus is reproducing and you're getting symptoms, whether it's an immune response symptom as well, but the virus is reproducing. Yeah. All right, let's find out.
Are we at the 15 yet?
Oh, we're 493. I'm not stopping until we get 500, folks. Bill, thank you so much for your contribution. Really appreciate your support of science communication at Microbe TV. Do your cousins call you Vinny? I haven't talked to a cousin in ages. Who knows? 496, 4 more. I'm going to let Amy go, and I'm going to do an after show. How's that?
I'm the big drawer, not you.
Well, let's see. We're going to let you go, and then I'll stick around. We'll see how many people remain.
It says 497. It's that I already missed those 499.
I'm just going to see. Oh, Ian, thank you so much for your contribution. Yeah, 500. We hit 500. Thank you, folks. I say,
really? You're at 500 likes?
Yeah, 500 likes. This is awesome. So now between that and my age, I'm a happy man until tomorrow. How's that?
No,
Siddhartha.
You said a month.
No, you said a month. Siddhartha.
And you agreed.
Okay. Thank you for your support. Gabriel, thank you for your support of science communication. 1,000 twivs. All right. There you go. Okay, Amy, thank you so much. for joining us. Why don't you go and
Okay.
eat dinner, and I'll close down, okay?
All right. Have a good night.
Bye-bye.
Bye.
Okay, let's do an after show, okay? Anyone want to stick around? Amy said everyone would leave when she left. Let's see how the numbers go down. I can go back and check out some of the, unless the mods want to go. Hey, mods, tell me if you want to leave. Just tell me, and I'll stop it. Because Amy left, and I can play here. I don't mind. Mods, you can go if you want. Everyone's saying goodnight to Amy, but I'm still here. Let me go back and see some of these questions that we could answer. Here in Brazil, we received CoronaVac and AstraZeneca. Then later they were Pfizer. As far as I know, showed it was more effective. Yeah, I'm still here. I'm going to stick around for a while if you guys want me. I'll keep answering questions. I wanted to let Amy go because she's got to eat dinner and she came right from work, but I'm good. I once did a 3-hour stream. Do you remember? Okay, regarding China, their vaccination rates are higher than ours, except in the elderly who distrust pharmaceutical medicine. Bodily autonomy is different than cooperating with social controls. It's interesting. So I thought bodily autonomy wouldn't hold in an autocracy, but it's out of my lane, right? What do I know? Listen, one other person are the only times I ever turn on YouTube. We don't monetize with ads, and that's it's a terrible way to learn, to put ads in. And most people need to have ads for revenue, but we leave quite a bit of money on the table by not doing ads. We depend on your support, and that's why we need you to give some money. Now we have more likes than people who are here. That's pretty funny. That's very funny. Chinese public health people also say that some elderly are too frail to be vaccinated. Yeah, I don't know what they mean by that. We vaccinate people of all sorts of ages, right? And Our World in Data, well, that's a good site for looking at vaccination rates. China vaccination rate is 89%, while the U.S. is only 69%. So it is not the rate, but the non-mRNA traditional vaccine. As someone said before, they mainly got 2 doses, which doesn't give you enough protection. Maybe that's part of it. I think the inactivated vaccines are simply not as immunogenic. I think that the mRNA vaccines would have been better, but they chose not to use those. And then they have the so I think 89% 2 doses is not good enough. And then a large fraction of the elderly. I think all of that plays into it. Oh, let's hear from some people who only a few people left. That's pretty impressive. There are levels of authoritarianism. In North Korea, I bet everybody got the vaccine. Yeah, there are levels. That's a good point. Are there any studies out there of how protective the original series are after 2 years? I think you could find some people, but it'd be hard to get the diversity you need for a good trial, right? So I'm not sure we'll ever see that. Yeah. This is the football play. The hit was not that severe, but possibly timed perfectly to disrupt the electrical rhythm of the heart, causing AFib. Okay. Prevent is considered by many to be an absolute. When describing vaccine effectiveness, probably best to say they reduce the likelihood of severe disease and death. Yes, I totally agree with you. And we should not talk about preventing infection, but unfortunately a lot of people have done that, right? And but I think you got it right here. Reducing the likelihood of severe disease and death is never 100%, right? And so people get vaccinated, get the full course, and then they get infected and die. And they say, look, vaccines don't work. No, it's not 100%. It's never 100%. We're very lucky that we have vaccines that work this well. Ooh, look at this. Wiz Khalifa performed black and yellow at the Dick Clark ball drop. And my renter mentioned it made him think of Racaniello after hearing him explain that's how people can. That's funny. That is very funny. Your renter. Your renter. That's really funny. Some argued that anosmia from COVID will give the infected higher antibody, which I don't believe in. The latest to have also shared the latest study, but any other thoughts? I don't know why it would give you higher antibody. I think the sustentacular cells are damaged. And there's a recent paper, which we'll probably do on immune, where there's continued inflammation of the olfactory epithelium, which doesn't subside, and that probably is also contributing to the anosmia. So we're starting to learn about what's going on there. We don't know the reason for that, but I think that's part of the story. Should people be just as concerned about a possible co-infection with Legionnaire's disease, bacterium, and coronavirus? Well, you would not want to be co-infected. I totally agree. But I'm not hearing reports that this is happening so often. The reason I paused is because Daniel Griffin asked, I asked him, has Africa been untouched? And he says, certainly not untouched, and hard to get solid numbers on how many died. Why do you ask? I asked because you guys here asked, so I passed it on to Daniel. So we can have Daniel on this show. I'll just text him your questions. How about that? Criticisms of the Chinese vaccine is also based on early efficacy with neutralizing antibodies. Do we know anything about the breadth of B and T cells? I think there have been some studies. I'd have to look it up because I don't keep all of the data in my head. I just know that they're there and I can look it up. And, unfortunately on a live stream, not easy to go and find things, especially if I'm here alone. But certainly it's out there. Rima, you're getting more stickers. You send me an email, so I'll sign the envelope. Yeah. Thanks, Frank, for moderating. Frank left. I think we have some others still here. I think Les is still here. Tom is cooking. Anybody has to go, it's fine. No problem. I'm just going to take some questions until we run out of steam. I would like a signed envelope with a random viral sample. That would make Antique Roadshow more exciting. So Vincent at microbe.tv, send me your address. It's funny. Someone sent me their email. Here's my address. I said, no, I need a physical mail address. And so they sent me a different email address. I said, no, I need the post. I don't know what to call it, right? Because if you just say mail, that could be email. But finally, I got their physical address. Turns out they were in Turkey. Okay, the Defunct Right did a job on the IgG4. That's great. Good to hear that. I will work this into my repertoire. That will be a much quieter option. Yeah, I'm not going to say it because now that Amy is gone, I don't want to get demonetized. Yeah. Yeah, this is a good point. Some people will die because of the vaccine or the disease after healing, right? If you vaccinate 8 billion people, one will die because of the vaccine. Well, yeah, sure. Or more than one, because we now know that men of a certain age, given the mRNA vaccine, develop myocarditis at a low frequency, lower than infection, but low, and some of them may die. No pharmaceutical, no biological product is without side effects. But the side effects are always less than the actual disease that you're trying to protect, right? Will the natural infections with the same intervals of vaccination do the same trick, or which is better? Well, that's assuming we put aside negative impacts of COVID. We can't, because you could die. So you really shouldn't discuss it. But I feel that the data say that similar intervals of vaccination or infection would give you the same outcome. Yeah, but you don't want to do that. I'm not condoning doing that because that will kill a lot of people. Now, physical addresses used to be called snail mail. I remember that. But this person in Turkey, I didn't want to use snail mail because they might not understand it, right? So post office, I don't know if post office would work too. Post office address. Okay, the MedCram paper, I'm still thinking of post office, was only 30 patients. Okay. I'll take a look at that. I will take a look at that. When will you explain how to get the 1000th episode tickets? I think we did, but if you want to come to the 1000th episode, you could start right now, and you guys are getting it first. I'm going to just send it here. Vincent at microbe.tv. And I bet we get 50 people out of that. And if we get just 50, you can come because I think we have 50 chairs. And if we get 100 people, I doubt we will, we'll do a raffle of some kind. Yeah, that's how it's going to work. Vincent at microbe.tv. People are talking about falling through the earth. Pescatarian is correct if you don't eat any meat but fish and also eat dairy. Yeah, that's what I am. I eat dairy and fish. Yep. Everyone's talking about passing through the earth. Bivalent does work at what, Evan? What does it work at? What are you talking about? Bivalent for 3 weeks has been shown to decrease infections, increase positives, because the antibody levels have been boosted. And it's compared to a historical sample of people who didn't get a bivalent but just got a regular booster done a long time ago. And that's an inappropriate comparison. And I guarantee you, if you go 3 months out, you will no longer have that protection. So there is artificially short-term protection, and it would have happened with the original booster as well. You don't need a bivalent for that. And that's the flaw in that. So don't say it works, okay? Doesn't work. Maybe you're not saying it works, but it's okay. Why would you fall past the core? Exactly. I love this discussion. I'll read it later. Gravity changes depending on how far you are from the center. A hold to China, you'd accelerate all the way to the center and then decelerate up to China. Okay, thank you very much. I learned a little bit about that tonight. That's pretty funny. I recommend you have people give you an email subject like 1000. Yeah, okay. So you send an email to Vincent at microbe.tv. That's less. That's a good idea. And put 1000 in the subject line. That way I don't have to search through them so hard, right? So it takes so long to do that, right? People are saying the variant in China is a recombinant. So the one of the X whatever, we went over it in the A&V papers, is a recombinant between two Omicron variants. That's correct. So for that to happen, two different Omicron variants had to infect the same cell, and they exchange bits of their RNA, and what comes out is a recombinant virus. Yeah. You bet. Although John says data, China has BF7 with BA5 mostly, so the recombinant is not BA5, so they would not have a recombinant. It is Elise, but it's pronounced Elise, short for Lisa. That's what I thought, because Lisa shortened to LIS is Elise, and that's beautiful to explain without having to come to the 1000th. At least we don't have to wait that long. Thank you very much. Lise. I varied between Lise and Liz, but that's good. Thank you very much. And one of the things I really like about this group is there's a lot of camaraderie here, right? We still have 470 of you. Okay, I did the experiment. 40 of you stay only for Amy. That's not a good experiment, right? Here's Elise. Here's a paper from Singapore, comparative effectiveness of 3 or 4 doses of mRNA and inactivated whole virus vaccines against COVID infection, hospitalization, and severe outcomes. Okay, so that's good. 3 or 4 doses. I'm going to look at that. If anyone's still doing stickies, please write this in the sticky. Les, I think you're still here. Tickets to New York have been reasonable. A live show would be a great excuse. So, another possibility is I get a bigger venue. So Amy found a venue of 52 seats, which is a nice little cozy thing. But maybe we get a bigger one if there's interest. I just don't think there's going to be a lot of interest, that's all. But maybe I'm wrong. Are you retiring from Columbia? No. I closed my lab this summer, but I'm keeping my appointment, and I'm teaching my virology course in the spring. It's going to begin in a couple of weeks. I don't know how long I'll keep it. I'm not going to keep it forever, but I would like to get Microbe TV really up and running and humming with a lot of people, not a lot, with a bunch of people working with me and well-funded, and then I would consider retiring from Columbia, but not right now. No. Not right now. Episode 1000 is going to be in April. Maybe the first 2 weeks of April. Okay. So you could write that down. We still get more likes. We have 530 likes. How about that? Some papers say that green tea, ashwagandha, and licorice root may be antivirals. So the thing is, the way you do this, you can grind them up and put them in antiviral assays in the lab, and they may have some antiviral activity. But then you have to purify the active ingredient and synthesize it and show that it's potent. And most of the time that's not done. In their original form, in the tea, for example, it's not enough to make a difference. That's the problem. You have to make it in concentrated amounts and show that it's safe and so forth. That's why a lot of these traditional medicines do work, but they're not potent enough. We need to get Vincent a bobblehead doll for 1,000. That would be very much fun. That would be cool. Now, you couldn't hide that from me, but it would still be cool. Your YouTube channel has 14 million views, getting close to 15. Oh, that's interesting. I would like to get 1 million subscribers, but I don't know if that's going to happen. We could certainly get 200,000 within the next couple of years, but gee, 1 million would be cool, right? We have to do something different, obviously, and that's not what we're doing now. Can I show you a picture of your dogs? All right, next time. I don't want to dig through it now. But the stickies, write down, please, get a picture of my dogs for next week. Yeah. More. That's why the nidovirales the genomes are so long, because they have proofreading capability. The nidovirales is an order, which is higher than, order is a collection of families. So not Nidoviridae, but nidovirales, which contains the Coronaviridae and another family as well. Amy had an Italian coffee maker. I don't know. No, we never had an Italian coffee maker. No. I mean, I'm an Italian and I'm a coffee maker, but I didn't make coffee in the lab, that's for sure. I read that a recombinant may have emerged from New York. So there is a recombinant, as we showed in that preprint. I don't think it emerged from New York. It emerged from somewhere around India, as they said in the paper. Please explain more on the furin cleavage site. All right, so the spike protein of SARS-CoV-2 and other coronaviruses has to be cleaved, physically cut by an enzyme in order for it to be active, to trigger fusion. And for SARS-CoV-2, there are actually 2 cleavage sites. And one of them is a furin site, and then there's a second by a different protease, and they both have to be cleaved. And furin cleavage sites are not unheard of in coronaviruses, but they're rather rare. And the furin cleavage site puts the protein in a conformation that makes it better cleaved by the other protease. And if you go back to the TWIV with Gary Whitaker at Cornell, he talks all about the furin cleavage site. And in fact, he took the furin cleavage site and put it in a different coronavirus. I think it was a feline corona, and it did nothing. He said, so the idea that someone just plopped this furin site into a virus and made SARS-CoV-2 is absurd, because you just can't do that. I think that was a great point from that. And that's something I should really pull out as a little snippet, right? And make a little video saying this is why it wasn't possible to engineer this virus, or one of the reasons why. Look at the number of visitors is going up. That's cool. All right, now I have to look up, what is the recombinant Omicron? XBB is the recombinant, okay? That is the recombinant. Is it, because you're talking now about XBB 1.15. I don't know if it's the same one. Because Siddharth is saying XBB 1 has these changes. But just, I don't know if XBB is different from XBB 1.5, because XBB is a recombinant. So if this is derived from XBB, this could be a recombinant as well. Virology Live covered Volume 1. I thought we did Volumes 1 and 2 in Virology Live. Have you ever thought about having an anti-vaxxer? We have. We're not going to have anti-vaxxers because they have no science to talk about. They're all wrong. There's nothing even credible about their science. So no, we're not going to have them on. I'm sorry. I hate to disappoint you, but we're not going to do that. I'm amazed that you guys are still sticking around. What is it? Should I do this every week? Should we do a post show like this every week? That would be fun. I don't mind doing it. I'm good. What is it called? An after show. There you go. Yeah. Richard, after show. Some streams do this, right? They just they say goodbye and then they keep going. So it's good. But we need to have some questions, right? Good. Many people are sticking around. After show is cool. Dr. Sellard. Failed measles contagion and has failed chickenpox contagion. I don't know what you mean by this. I don't have enough data here to make an intelligent answer for you. I'm sorry. Yeah, on the West Coast, it's early, right? Yeah, this is the end of the Marvel movies. Yeah, but this is not as... Remember that one where they're sitting around having dinner and they're all tired? I like that one. I think we should do that on TWIV also. We're going to have a live stream TWIV, as I said. And yes, I think Amy was great tonight, right? I think I didn't do anything untoward, right? Which is important. And she was in a good mood. So yeah, she was really good. I liked her. Comparative effectiveness of 4 COVID vaccines against B1.1. This is Omicron. I saw this paper. It just came out recently, and we're going to take a look at that for sure. Yeah. I can't do it now because I haven't read it now. Tell us some gossip. No, I wouldn't do that because she may be listening, you know. Right? Amy could be listening, and I don't want to say anything. But even then, I wouldn't say anything that would offend her, or I don't have any gossip, actually. I've been a fan of Microbe TV since 2017, but I think Twin needs a B12 shot. So what does that mean? Some reinvigoration? Give me some suggestions. I like that the 2 youngsters came along, right? Tim and Vivian. I really like them. But if you think we need somebody else, let me know. because Ori's busy. He's a resident, right? So he's not always there. And he do developments on long COVID. I would say we're making slow progress. People are working on it. There's a lot of money put into it. There's some starts and fit starts where people publish stuff that's just not right, saying it's an infectious virus here that's persisting. No, it's an inflammatory disease, and we have to figure out what combination of cytokines is doing it and what's triggering them. And I think we'll get to it. It's just not going to be overnight, right? Vaccine is better than infection. Yeah, a vaccine is better in the sense that you don't die most of the time. Right? You may die from the vaccine, but very rarely, much more rarely than getting infected. And it's a controlled immunization, right? But in terms of an immune response to the natural infection is better. You're getting a response against all the viral proteins, right? So I'm not sure you should say that immunologically the vaccine is better, but certainly it's safer. And so we should all be vaccinated for sure. Why are there no more virology courses? Well, because there are not many virologists. Most universities and colleges do not have a virologist. And so it's hard to teach a virology course. And I know a lot of people who use my lectures to teach their virology course at some small college or university, because you can't just pick up a book and learn virology and then teach it. That's not good. The best virology teachers are people who have been doing it for a while. And so they can use my lectures and then have a discussion. I think that's great. Or they can use my lectures to try and learn. But that's the problem. And a lot of colleges now say, oh, teach virology to someone who's never taught a virology course. I don't think that's the optimal solution because they don't know virology. Now, they should clone me, I guess, but the lectures are all recorded, so they can use them. They could assign them to their class and then have a discussion afterwards. I think that would be very cool. So that's the reason, because there aren't many virologists, and I'm a big proponent of experts teaching, in their subject matter expertise. And I'm doing my virology course now in a couple of weeks at Columbia. All the lectures will be recorded. They'll be uploaded on YouTube so everybody can learn virology. You graduated. What are you going to do next? Let us know. In the U.S., what do you think is the percentage that have had no vaccinations and haven't had COVID? It's less than 10%, I think, right? The total immune from vaccines and infection is over 90%. So that's not bad. So you bought the ASM textbook, Principles of Virology, right? Good. Media still did and still does a horrible job at educating the public about what vaccines can and cannot do. They do. They don't talk to the right people. We try and help, but we're small. They don't listen to us, et cetera, et cetera. How many Twix hosts have been on Jeopardy? Only Brienne, as far as I know. Yeah, Brienne is the only one. I'm not going to be on. I could never do that. Oh my gosh. A twin host was on Jeopardy? Who was that? I don't know of that. I'm not aware of any twin host. I just wanted to see if Daniel was responding. Someone said, turn your phone off, Daniel, but I don't think he will do that. He needs to get text from the hospital, right? Okay. Why are doctors, virologists all over the world claiming that many people get sick or die because of the vaccine? They have misinformation. They have anti-vaccine sentiments, which means they get paid to do that. to be anti-vaccine and promote their alternative stuff, or they don't understand. They're just wrong. I don't know why they do it. I think there are a variety of reasons. But yeah, that's not true. It's absolutely wrong. And I don't know why people love to focus in on what's wrong and not what's right. So we have a minimal audience, right? And the wrong stuff gets millions of people listening because, I don't know, because they tell people what they think they want to hear, I suppose. Send your postal address. That's a good way to put it. Send your postal address. So anyone who wants to get a sticker or anything else, Vincent at microbe.tv, send your postal address in that email, okay? They said no one has died of myocarditis. I'm not sure that's true. I know people have died from blood clots. Remember that with the AstraZeneca? Let's take a look. Myocarditis, deaths, COVID. I'm going to get all the anti-vax stuff right. This is one study in JAMA, which is in January. It was a year old. In that study, nobody died. But, there have been subsequent studies. Mortality higher in myocarditis after virus than after COVID. All right, so according to that, there have been some deaths after vaccination, yeah. Oh, I remember a parent saying a baby was watching TWIV, although they might not understand the word, orama. Yeah, I think someone said their young child kind of focused on it for a minute or so and then went off to do someone else. The address where you live, postal address. Clusterfest has no expletive or derivative within it. Yeah, it's true. Shipping address. I'd be surprised at how many people would come. Surprise me. I'd love to be surprised. And send an email to let us know your intention, because if a lot of you want to come, we'll get a bigger venue. And we're going to have the whole crew there, right? Including Daniel, of course. And what exactly we do is the other issue. I'm not sure. I don't want to just do a paper, right? We have to do something interesting. Maybe we need to have some cool guests, right? Yeah, we'll see. Come to New York City in April. Yeah, it's going to be April. I can't delay it more than that. XBB is BJ1 and BN1. Are you wearing a mask when you shop? I do not wear a mask anywhere any longer. No. I wish we could find out why some people have minimal symptoms while others die. Most illnesses are like that, right? And I think for viruses anyway, a really good interferon response is a big part of having minimal illness, but it may not be the only part of it. Is it true that Omicron infects faster than the antibody response? This is one of the ideas out there. It's anecdotal, I think, and no one has done the right experiment using infectious virus assays in people to be able to address this, right? You'd have to infect people. So I don't really buy it. Parosmia, there's some people who say that they can help, but I'm not aware of it. People say that you can retrain, and there's some people out there that have success doing that. I'm not familiar with that, though. Maybe you should write a letter to Daniel at microbe.tv. He may be able to answer that. I listened to a talk by the author of the paper on autopsies. He did discuss preserving samples. He had to get up in the middle of the night to do so. Well, hopefully he will do that. Paxlovid rebound is not a thing. It's COVID rebound. You will get a rebound of COVID in many cases, with or without Paxlovid. It's a natural part of the disease. You have a viral phase, the virus goes down, and then you have an inflammatory phase, your immune response causing the symptoms. That's the rebound part. Traveling from Central Texas, wow, you just hop on the plane with Rich Condit, right? Go to Austin and come with him. Try to get Miller Theater at Columbia or Low Library. Yeah, those are good ideas. Maybe they would be preferential to us, right? That's a good idea. I'm going to write that down. Very good. And I'm not sure we're going to have an open air option because I just can't be outside, right? Sorry about that. Has there any been data showing why some people can spread COVID better than others? The only data I know of is that they make more viral RNA. So, a certain fraction of people are the ones that make the highest amount of virus when they get infected. And so I think those are the ones that transmit the best. But I think it's certainly not a resolved issue by any means. No. Nope. In the A&V papers, would you consider putting a note to say which papers are or bad? No, I don't want to do that. Let people do it for themselves. But, here we're going to do that. So people could, maybe we put a note. I don't want to do any more notes. It's too much work. I have enough work as it is to add things here and there. I do the A&V papers myself. Maybe someone would like to do them. Someone who's already in the website. Who knows? Why are youths dying globally in excess? Not just youths, but the paper we did before is there total excess deaths across the board because people don't always put COVID as the cause of death. And COVID can induce deaths, respiratory failure, heart failure, etc., and they're not ascribed to COVID. They're indirect. Even psychological deaths, suicides, for example, people freaked out because they don't have their lives. Those are all COVID related. So there are a lot of excess deaths, and this happens with every major infection, major epidemic. Did everyone die before antibiotics? No. There are always some people who survive infections, but many people do, and it's a horrific death. So you want to take antibiotics. That's why they're useful. The same thing with vaccines and antivirals, right? Egypt reports on genetic variations on human beta defensin 2 and COVID outcomes. So this is sort of expected, right? These are peptides involved in antimicrobial defense. And so we're going to be seeing lots of genetic associations with these defense proteins and severe COVID. But it's going to take a while for it to sort out. Yeah. Original antigenic sin means that you have, let's say you get infected with the virus A, and then you get infected with virus B, which is related to A. Your immune response is mostly going to be against A. The memory response against A is going to be very little response to B. That's original antigenic sin or imprinting. And so HPV, some people have found that you get certain serotypes in your HPV vaccine, and then later you get a HPV vaccine with more serotypes, but you don't make a great response to those new serotypes. You make mainly a response to the original serotypes that you got in the first vaccine. So obviously that's not good because it's the same thing with the bivalent boosters. When you get a bivalent and you've already had the original vaccine, most of the response is to the original. It's not to the bivalent. It's very low. So that's imprinting. Same thing. Virology Live covered both volumes. I thought so. Yeah, for sure. We're just getting to the point where people are saying yes to an after show. I always wondered why Vincent didn't stick around. I don't know. I didn't know if people would stay. I mean, the mods are sticking around. They've been here for 2.5 hours now. We'll stop at 11, okay? I don't know. I'll make it a thing, okay? I will make it a thing. When Vincent gets tired, let's bar crawl. I'm way out in the burbs here. There's no bar to go to, but I would love to. Oh, it still says Amy on the screen. Oh, my gosh. How do I get rid of that? No, that's not it. Where's the Amy thing? Right there? Nope. I just have to click on it. All right. First, let me get rid of the thing, and we will get rid of that. Okay. So, hey, I'm sorry. I didn't even notice. It's okay. She's here in spirit, right? Yeah, variants. Do not worry about the variants until someone shows that the vaccine doesn't protect you against severe disease and death. I don't care about neutralization titers at all, because they've gone down with variants before, and we're still well protected against severe disease and death, as Paul Offit says. So with the current variants, do not worry until we see that there's an uptick in severe death, okay? It's not severe death, severe disease and death. And the study has to be done properly, right? If you study the wrong population, it's not going to give you the data that you want. So don't worry about the variants. Certainly don't listen to the press talk about the variants. Don't listen to oncologists talk about the variants. Listen to virologists. I'm not the only one, but most other virologists don't talk like this. They don't interface very much. Hey, look, we're here for 3 hours a week. Nobody does this. They're out of their minds. Amy is watching while having dinner, you think? Okay, people want to say hi to you, Amy. Amy, they loved you tonight, okay? Very good. I don't think it's stressed enough that SARS-CoV-1 and MERS both originated at markets. I keep hearing people pushing lab leak. Well, MERS originated from camels, not from a market, okay? But SARS-1 certainly did. Yeah, in history, John says that every virus has come from animals, not from a lab. And so history is often strong and a good reason to think something happened a certain way. But on the other hand, look, while we have to be aware of history, we can't be completely hamstrung by it, right? We have to admit that some things can change. So, okay, that's fine, but there's no evidence for lab leak. I don't know why they push it, because we don't know where it came from at the moment. There's no, 100%. Look, it's clear that, to me anyway, that the market was the origin, the Wuhan seafood market, Huanan seafood market. But we don't have the virus isolate. We have other kinds of data to prove that. So people say, oh, well, you don't know, so I'm going to say it's lab leak. So I think that's all that is. But absolutely, absolutely not. I'm going to be involved in a documentary about this, being made by a UK company. So they're going to talk to me about it. And they want to have both sides of the lab leak, that it came from a lab and it didn't. And I don't think that you need to do that, because there's no data that says it came from a lab. All the data says it came from nature. So anyway, I'll be on that. They're coming to the incubator to film it in a couple of weeks. Do we know why varicella zoster manifests as shingles later in life, but not during the initial infection? No, I don't think so. But, initially the virus is coming in you. It's respiratory transmitted, so you're inhaling it. It's reproducing in your respiratory tract. And then it's getting into nerves. And so there's a viremic stage, and the virus goes to the skin. You get the rash. Some of it goes into nerves. So then the reactivation starts in the nerves. And then the nerves innervate certain parts of your skin, and the virus goes from the nerve to the skin, and you get a rash along that nerve, the dermatome controlled by the nerve. So I think part of it is how the virus is, where is it starting? And that plays a role, but we don't understand the whole thing for sure. Oh, thank you. We stick around because of you personally. You're a delight to listen to. Thank you very much. It's very kind of you. Bring on the salty lingo. How long do we now think immunity lasts following an infection? What are we talking about? SARS-CoV-2, right? So I think the course of this pandemic, we still have immunity, right? We haven't exhausted it yet. So 3 years. I don't know how much longer it's going to go. There are T cells left over from the original SARS-1 in people for 17 years. Memory T cells. So it's possible that memory T cells could last that long. But this is a different virus, and we don't know. And we don't know about memory B cells. So we'll just have to see. But at least 3 years now, maybe 4 or more. We'll see. All right, now I've seen the core a million times here tonight. What is this core? Is it a good movie? Should I watch it? Because remember, if I watch it, I'm going to do less side communications. Boosters don't prevent infections, okay? They, for a period of time, boost your antibodies, maybe a few months, prevent infections. But then the antibodies go down and you get infected and you get symptomatic COVID. And it's not a public health strategy to keep boosting every 6 months, all right? It just is not. So we cannot justify that. That's the end of the story. You can't use another teacher's lesson plan, but someday you could use my video and let them watch it and then make your lesson plan based on that. A lot of people do that. I don't see why that's a problem, right? Who owns your virology lectures? I do. They have no problem with me putting on YouTube and doing whatever I want with them. I checked that years ago when I started teaching. I said, is it okay to put it on YouTube, Coursera, a bunch of iTunes? I put it on for a while. No problem. Do you think the mRNA vaccines will be coming out soon for flu or other uses? Maybe. They're being tested. I don't know how that's going to turn out. We're going to have the guy who did the flu mRNA vaccines on TWIV hopefully this month. We'll see what he thinks. But I think they're promising, but we have to see, right? Do you have an online virology course suitable for beginners? Not yet, but I've been asked to do this, right? Basic virology for basic people. It's a name that someone from the UK suggested. It's a good idea. He sent me a physical letter, a snail mail letter. Basic virology for basic people. So I'm going to do that in the next year. I wish I could do it just like that, but it's going to take some work. So stay tuned. Yep. You still want the textbook. You don't have any copies of the textbook, and you don't care if it's an out of date. So send me an email, vincent@microbe.tv, Questella. We'll work something out, okay? Yeah, this is office hours, for sure. It's just two virologists chatting with you guys, and that's what we do at office hours, for sure. How do you deal with future when public is so divided about mRNA science? A lot of people in the U.S. at least got mRNA vaccines, and in many other countries. So I don't know if divided is the right word. There's some dissension. But I think with time, as safety is shown and as other mRNA vaccines come out, there'll be less resistance. And we have to have good communication from sources like this that show you, here are the data. This is why they're safe and they work. Yeah. Would the 2023 lectures be on YouTube? They certainly will. They're going to be recorded because I teach in a classroom, and it's not easy to do a live stream, unfortunately. But the class starts the week of January 17th. Something like that. Let me just tell you exactly when. First class is Wednesday, January 18th, and I'll be posting them on YouTube within a couple of days after that, yeah. But I don't think I could stream them, no. I can't do that. Saw this on the Telegraph. Use of fear to control behavior in the COVID crisis was totalitarian, admit scientists. I don't know what it means, actually. But that's interesting. I would like to see that. Vaccinations, I look at it as a huge bell curve. The huge portion in the middle are us. We are the fortunate ones. The immunocompromised are at one end, severe reactions at the other. Yes, everything is a bell curve. Well, maybe not it's a bell curve, but everything has ends, right? And it's never black and white. Biology is not black and white. That's what I always say to people. I just am amazed that there's still 462 people here. Wow. So 462 people for the after show. I only look 60, and we had over 500 likes. Now we have 570 likes. Wow. Maybe you get a different crowd as you go into the wee hours, right? So if I went to midnight, I get a different crowd, and then after that. But I don't think I should do that. TWIV 1000 t-shirts for sure. We have to have a cool design. Maybe we have to have a contest for a design, right? Yeah, that would be cool. What if for TWIV 1000 you create a Jeopardy type game with categories? That would be cool. Now, but who's gonna participate in this? Am I going to be the guy, the Alex, whatever his name is, and the other panelists are going to answer? That would be cool, actually. But Brienne would win, right? That's a great idea. Get the guests to share how TWIV has affected them. Yeah, we could have guest involvement too. I think that's a good idea. All right, so stickies. Guests to share and a Jeopardy-like thing. We could do several segments, right? We don't have to do all one thing, for sure. I don't know if you saw 500 in Austin. We had 3 guests, one after another. We had a pro-vax lady from the area, and we had 2 virologists come on. That was good. That worked well. We had them each on for like a half an hour. So we could have also a local guest or two, right? That would be fun. In addition to a Jeopardy and a hearing from the audience, we could have some local virologists. That would be cool. Yeah. Can you have pneumonia without knowing it? Yeah, you could. You can have what's called a walking pneumonia. It's caused by typically bacteria, where it's essentially an asymptomatic infection. You don't feel anything, right? Is there a genetic component to infection immunity? Absolutely. It's the biggest part, right? There's the virus and then there's the host, and the host provides a huge genetic component. Every human is genetically different. That's why we have a huge, varied response to all kinds of infection, from no symptoms to death. That's because we're genetically variable. And to figure out what are the genetics, a lot of people are doing that. It's really interesting. And we have some clues for some viruses. For herpes viruses, we know what causes encephalitis, leads to encephalitis. We're getting some clues for COVID as well. It's really exciting. And those things I'll talk about in my course. Apropos people who do the most transmitting, it doesn't correlate to severity of their own disease. No. So they can make a lot of virus and transmit well, but they're not very sick. Yep. Absolutely. The vaccinated will make less RNA, so will transmit less, and boosted will make less RNA. Well, the boosted will make less for a while until the antibody levels decline, and then they'll make more, but not as much as someone who's not been vaccinated. So vaccination or infection does reduce transmission, doesn't eliminate it, though. It does not eliminate it. I don't know the answer to this, but maybe someone can help. The best book for laboratory hematology. That's not my area, so can't help you with that. Here's a link for the Virology live course. Who is my favorite Nobel Prize winner? Oh, I'd have to say David Baltimore. I worked with him. And I think he's a terrific guy and really smart scientist and helped my career very much. So he'd be my favorite. Well, Bad Cat, that's simply wrong. You're just saying something that isn't true. And there's plenty of evidence for natural origin. You just don't want to see it as such. But I see the data and you don't. I'm sorry about that. Nature is a staggeringly huge lab. That's absolutely right. Okay, let's get down to the business, to the bottom here, because it's now 10:55. Yes, the course is great. Vibrio is a type of bacteria that lives in the ocean and can periodically infect people and cause cholera. Yeah. I've read that wild circulating chickenpox among children used to remind the immune systems of older people, thereby suppressing reemergence as shingles. I don't think so. We had to vaccinate to suppress shingles, so it doesn't do that because it's latent in nerves, and so immunity is not going to help. I haven't seen the core, no. Should I see it? Okay, I'll see it. We'll see the core. It's so good. All right, no problem. It's awful? Someone says it's awful. Okay. Movie opinions differ. That's true. I don't get a chance to watch live streams, but I watch afterwards. Well, thank you. Thank you for watching at all. It's great. And this is a lot of fun. I love doing this, and I could do this for a long time. Pfizer phase 3 mRNA flu trial will end in mid-March. Okay, looking forward to that. We'll talk about it. We'll talk about it here. Yes, you can buy an out-of-date textbook online for sure. For sure. This year, we'll do it this year. No problem. Am I willing to entertain that crowd? The TWIV 1000 for sure. All these things that we've talked about are great ideas. We're going to do it. And the crowd gets crazier after dark. TWIV After Dark. That's another name. We have, what was the first name? It's After Dark. It was always After Dark. It's a good name. Yeah. If you told me you were mid-50s, I wouldn't think you were lying. Okay, we need a T-shirt design. I'll take polio for $200. That's great. I love that. We're going to do that. Write these down, sticky people. I'm going to go 3 more minutes to hit 3 hours, okay, because I want to hit that 3-hour mark. Do you think journalists deliberately pick non-experts to get drama? I don't think so. I think there are certain people, I call them pundits, who are very visible, and they'll get interviewed, and then they make a splash, and then the press just keeps going back to them, and they're not the right people. The right people don't talk to them. So I don't think the press wants to get drama, although they do end up finding people that make the drama. Yeah, for sure. Yes, so besides people being super spreaders, there are venues that have super spreading events, right? But there has to be someone in there who is shedding. And I think if this were another night of the week, we could have cocktails. But I'm just drinking water because I want to stay fresh for tomorrow. I got 2 pods tomorrow, right? We have, that just one? I guess tomorrow's Thursday. We have Daniel tomorrow, and then Friday we have TWIV. Are there any dangerous side effects to immunocompromised individuals when taking the mRNA vaccine? Not that I'm aware of, because the only problem is that they don't respond well to the vaccine. You have to give them multiple doses, but it's a non-infectious vaccine, so there are really no issues there. I guess you're liking After Hours. This is great. We're almost at 600 likes. I don't know where they come from if there are only 436 people. I guess people come in and out, right? Could we get to 600? 585, 15 in the next 1 minute or so. After midnight. Yeah, last call. This is great. I love it. Okay, folks, last call. with cocktails. You guys are keeping me going here. I would argue the press wants all the drama. To sell their product, they need to generate some interest. So if it's drama, but that goes for any kind of press, not just science, right? But science, you shouldn't do that because you need people to get the right information. It becomes Vinny after 10 p.m. You can call me Vinny. That's fine. Vincent or Vinny. I don't like any other version. Is there some info on Paxlovid helping for long COVID? There have been, I can't remember the studies, but certainly there are studies in progress with Paxlovid. I don't think we have a Paxlovid result yet. Remdesivir, we might have to ask Daniel about that. Okay, it is 11 p.m., folks. Let's call it a night. That was a cool, 597, 3 more. Let's do it, folks. We're going to stay here to get that 600 likes. Come on, there has to be 3 people here that haven't hit the like button. Could you poke it before we go? I'm going to sit here. Maybe it'll take me a 599. Very good. Very good, folks. One more. In the meantime, I want to thank the moderators. I don't know who stayed here. I think Vanity's gone, but I saw Les here. Tom, perhaps. But thank you all for being here. 599. You got 600. Yeah, I'm a little delayed on my feed. 602. Thanks, folks. Thanks, everybody, for coming. Thanks for sticking around. Love you all. Right? Can I do the heart? You know, the other heart version, too. That's pretty cool. Love you all. Really enjoy your company and your questions. Bring them back next week. Till then, stay curious. Good night.